It's okay. This is nice. Good morning, everyone. Um, Nobody recognizes me here because I'm not from your community, but it's a pleasure. Thank you for waking up early in the morning to join us. Thank you for Sebastian, Maple, and the amazing team, Joanne and Milea here from the previous workshop for leading this effort. It's really a community gathering effort where we want to bring people together to discuss this topic. Um, my name is Kedar. I'm not from NUS, but I also work in NUS. I have an NTU, but that's okay. I'm also an IMRI, an A-star. And I'm along with Professor Zhang, who's sitting at this table over here. And we're leading the AI for Science initiative. We're helping NRF essentially to lead the AI for Science initiative in Singapore. So it's a bit of an accelerated thing. This is how we tend to do things in AI anyway, right? But can you just flash my slides? We started off with a workshop back in February, which was introducing AI for Science in Singapore and the general initiative. Um, and essentially, I think there's one slide before this, but that's okay. Yeah, so yeah, thank you. So all the information is actually available on the website, aiforscience.sg. I'll flash that later to you. But essentially, we have many workshops in different themes. We're going to have one on healthcare, as Sebastian suggested. And so you can find this information here. And we're working towards having this conference called the Nobel Turing Challenge and AI for Science Conference. Next slide, please. And the idea actually came from Hiroaki Kitano. And this is sort of the epitome or the pinnacle of what AI for science would be is, can you have an AI system to give the Nobel Prize without noticing it's an AI system, not a human scientist, right? So we have to get there. So the AI for Science initiative is hopefully getting us to that stage. Next slide, please. So what is AI for science? I think it's important to sort of define this, and we've written this in the scoping report. So if you actually go to the website and you scroll down, there's a section called scoping report. This is the initial report that Professor Zhang and I wrote to actually describe this. So AI for Science, we define this as the general effort to utilize and extend AI technology for solving scientific problems towards scientific discovery, right? And so what we feel is there must be a very strong handshake between AI methods and scientific problems that domain experts such as yourself will define, right? That's really important to identify. Back Science of AI is a bit different. So differentiating is very important because there are many terms for science of AI, research for AI. What you'll hear maybe perhaps in the next few years is foundational AI development. So AISG is actually doing this quite well. And this is different, which is referring to the development and advancement of AI technology and algorithms themselves, right? So you do need this work as well. And we want you to work very closely with them if possible. AI for X is the implementation of AI to solve practical problems. There's quite a bit of this effort ongoing as well, but this is not what we would like to do. And this is really the purpose of this workshop is to define grand challenges in your domains that can be solved only with the help of AI. And that's really why we're bringing all of you together today. Next slide, please. Okay, so the way we're thinking about this, and this is very early thinking, is that there are various domains that have their own grand challenges. This is where all the thematic workshops are happening. So you could call healthcare a domain. You can call genomics a domain, for instance. There will be cross collaboration between them. That's completely fine. Okay, we expect that to happen. Okay, I don't know who's doing this. Okay. His request is not wrong. You can record the meeting. It's just that let's not make sure that pops up over. Okay, so there are various domain-specific things that are coming up from the ground as well. So I think this is something that we're working towards. We don't just want to be a directive from top down saying, "Oh, we love climate and health, earth sciences. Let's have a program on this." That's not what we're after. And there are, I think, enabling technologies such as data as well as computational resourcing, including hybrid compute, so quantum compute, for instance. So if you're able to define what are your data sets that are critical to solve these scientific discoveries, do they exist? If they do not exist, what effort is required to make it exist, especially in the Singapore context, connecting globally? Right? That's really what we want to define. And so data and compute are going to be absolutely critical to solve these grand challenges, right? So please do discuss this during the workshop as well. Next slide. Okay, so we're going to work backwards. This is actually where we are today is, all of these thematic workshops. We're hoping to finish them by the end of April, by May. NRF will hopefully launch the Grand Challenge call for proposals sometime in August or September, right? And the ideas that you form today, the networks you're creating today will hopefully prepare you to actually submit proposals when the call comes out. Okay. What we're working towards, Yang and myself, are the AI for Science NTCI workshop and a full study report, which will be a public document that will be disseminated to all the government agencies, regulatory authorities, funding agencies, and academics, industry partners. So it's available on the NRF website. Feel free to publicize that even globally, right? And all of your outputs from this workshop today, through Sebastian's help, will be a part of a white paper that can then describe your field, and we'll integrate that together and write up the full report. So that's basically Yang and my job. So we're going to finish that probably by the time the AF Science Conference comes around at the end of July, right? So we really need your help here to be as clear as possible and articulate your vision as clearly as possible so that we can actually put this as part of the FRC report. Okay, the next slide. So this is the website, as I mentioned, AIforscience.sg. I don't have a QR code, I forgot to do that. But we started off on the 5th of April, and we're going through all of these different themes. Please do attend as many of them as you can, if you can give the time, even if you can attend for part of it online, all the attendees online. Thank you very much for giving us the time. We're going to have some on chemical, biological manufacturing. There's going to be one on healthcare, which is not on the website yet, but we're working on it. Next slide, please. All the way to the 10th of May. So 10th of May is where we actually have a workshop on AI methods, right? And I see some domain experts in AI over here, and we will actually collect them together. And the grand challenges that you have defined in this workshop will be distilled, and hopefully Sebastian will present on that day to say, this is the problem in biomedical sciences, or these three are the problems. These are the data sets. These are the benchmarks. And then we will actually get buy-in from the AISG and AI community to actually say, okay, these are the methods that we want to do to solve them. Okay, so that's what we're hoping for. Next slide, please. Okay, so what do we want from the workshop today? As I mentioned, we want a clear vision statement, or multiple vision statements, defining the problem statements and set target. We need to understand the challenges and the bottlenecks towards solving these problems, obviously, right? Define how Singapore is unique in addressing these. That's important to actually justify in a way. And I think data. So I think we've seen the previous workshops as well. Maybe I can request you to kindly define very clearly what data sets are available and what data sets can you leverage on their public. What is the problem to accessing them? Or what do you want to develop in Singapore? You know, think big, please, right? Say, I would love to have 1,000 GPUs dedicated to do, something, cell level data collection. Please mention that, right? So bottom line, what we want is transformative ideas. Really, AI for science has to be transformative ideas, 10 to 100x improvements in something. Data sets that are available or to be curated, and HPC resources, GPU or CPU is needed. The more you're clear about what you need, like, we need these many resources to be put in, this much dedicated resource from NSCC, whatever it might be, please mention this, and we'll try to capture all this information, and then we'll work towards it. Okay, I think that's all I have. Okay, so the last slide here is what we're hoping for is that if you see, as I mentioned, this foundational AI research and this applied AI research, and AI for science sits in between. So what we're going to hopefully do is identify the sectors and the data, and then AI to provide the catalysis to do that. Here's an example for material science. So you have the foundational models, could be transformers, could be something else, right? A different architecture. And then you can actually have a particular problem that you can solve because of that. So we're working on this, for example, in materials. We actually tune the models such that we can solve the materials problem because they're unique situations how the existing models just don't work. So we have to modify them or make them better in different ways to do that. And then eventually that goes to your AI-guided design for something, right? In this case, it's a receptacle, could be DSO. In our case, one of the cases is DSO, could be a company, or could be another academic who's actually doing more translational research, right? So sometimes this question comes up. We're looking for very low TRL over here, right? If that helps you put things in perspective. We're not looking for TRL even 4 or 5. That's okay. We're looking for foundational grand challenge ideas that are enabled by AI for science. Okay, so hopefully that's clear. And I'll pass this over back to Sebastian. Thank you.
Thanks, Kedar. So again, Yang and Kedar are the leads for the AI for science and have asked us to cover different topics. And the topic questions. Questions, yeah, Sham.
Interesting discussion with someone from URC, not from East Africa, but I have. Oh, you are? Okay, sorry. Yeah.
So the question of, we were debating what is AI in this context, because, for example, in some presentation, we've seen that the vision is data, AI, compute, with these three interlocking gears, and so on. But from what I was given to understand is that their vision of the leadership's vision of AI is just whatever shorthand for AI plus data science plus algorithms plus data analytics, everything. Except that the word is a powerful word. There's a reflex to say AI means neural networks.
Sure.
Right. So I just want to clarify in the context of AI for science, does AI mean neural networks, or does it encompass all of algorithms and data analytics?
Yeah, so I mean, it definitely not restricted to neural networks. I think we've come very far from that because we've shown in other domains that you have to have architectures in the way you design an algorithm so that it can solve a specific problem, right? So one example that I can give you from my field is that you would take an existing architecture, it's still a neural network, but we build the architecture so it obeys the physics of that problem we're trying to solve. Okay, there are various ways to implement that through regularization, through the architecture change, and so on and so forth. And that's actually fundamental development because that field can only be solved through architectures such as these, right? So AlphaFold is another good example of this, and Yang knows more about this than I do. But just briefly, you have a particular end-to-end learning platform and including the physics in such a way that you're able to learn it, right? So AI is very broad. I will definitely not restrict it to any, just a neural network, but any one particular kind of algorithm. There's no restriction.
Can I clarify that even the examples that you said are not restricted to neural networks, are neural networks?
Oh, no. So, for example,
Right.
we use variational autoencoders. You can and cannot use neural networks in that case, right? It depends on what works best for you. So neural networks are good for large data because deep learning is powerful for large data, right? Which is why there's no better solution. But if you can articulate why neural networks are not the best solution in something else, absolutely.
Thank you.
Maybe important is that the outcome which you have this call depending also strongly on the reviewers. So the reviewers' understanding of what is AI is something that is important. So the profile of reviewers is also something that maybe we should add towards that. Because especially in the field that is across, you have biomedical people. For them, something else may be AI rather than someone that always goes to IEEE conferences, etc. So I think that's another thing that in that stage later should be clarified.
Thanks for that, Sebastian. I've seen these, I'm bringing this up insistently because I've seen these assurances before. But then as Sebastian said, if all the reviewers are neural networks, people who go to NeurIPS and take that as their definition of AI, they are not going to be cognizant of the reasons why neural networks may not be the solution for everything, right? And so de facto AI will mean neural networks.
Yeah. So I can give you something from personal experience. I can't speak for everybody, right? So some of the reasons why this happens is because, let's say there are grants where you're expected to deliver in the top computer science conferences. And of course, the people there would care about, the benchmarks in terms of the AI methods that are improved. In this, we don't care. This is really true. This is not just an assurance. We want to actually have Science or Nature papers or whatever come out of this, right? So it doesn't matter how you get there. We think our belief is that you can only do that if you solve the grand challenge in the field, which is enabled through AI, be it whatever form it might take.
Thanks so much.
So we do have a schedule to go through, but I also have to say that this workshop, I think the format is very interesting, and I'm also very curious how we're gonna manage it together. But a lot of credit also goes to Joanne and Mile. I saw how they did the AI for Genomics workshop, and we've done similar things and maybe just make a few changes to it. I also have to say we have 100 people online, and there were over 200 people registered, and was not easy to make very quick decisions. As you saw the short notice on also inviting you to come here in person. So it was really trying to get similar to what Joanne did. First, from every unique entity, have one representative, and then there are duplicates only when there are extra spots available. And then, of course, we also got our people to help. We have scribes on tables. We've assigned some of the topics that came together. In fact, after the first AI for Science workshop, there was a survey where people could choose what area are you interested in. So based on the frequency of what people chose, the workshops were chosen. And in the AI and biomedical science, the subtopics that were the ones most commonly chosen were AI in drug design and AI in synthetic biology, where AI in drug design had a much larger, double the amount of people interested. This is why there is a strong focus today on the AI in drug design. And we have representatives from all of the major clusters, universities that contribute rapid presentations. And I will just guide you through a bit of how the day this morning will work. We will also ask you and the people online, and I ask that you also post the Pigeonhole link online, please. So the way how we can try to get the online audience also to help is we want these challenge statements that are one of the outputs that we want to get in this Pigeonhole. So the Pigeonhole is not meant for chatting, voicing problems. You can still do that. But what we really want is some of these challenge statements of where AI can improve, create impact in the biomedical sciences field, including what kind of AI, what kind of data can be used. And then through the system, also you in the room here and everyone online can vote. What are the main topics? And our tables, they have some predefined subtopics, but we have extra tables that take the top hits from the Pigeonhole discussion. If we see the Pigeonhole discussion goes in many different areas, we'll try to also react to that and make sure everything is heard. And as Kedar said, we will bring to Kedar and Yang a summary of everything you're discussing here in this room, and also from the online discussion, so that we get a good spectrum of what is the interest and the need in AI and biomedical science. So maybe can you click on the presentation? So in this list of speakers, we will start going through first AI and drug design. We have some summary on synthetic biology, and we'll have a bit out of the order talk about the different sources of data. Because if you think of biomedical sciences, it can be many, many things. So we want to encourage that if there is a niche area, it can still be important, and I want the voices to be heard. So before going to our speakers through these rapid fire presentations, the format again of the breakout sessions will be, you want to identify the areas of opportunities involving AI to contribute towards biomedical sciences with great impact. And there are some examples here below. Don't be biased by it, but it's something like, how can you make drugs more affordable? How can you harmonize biomedical data accessible for AI? All in the topic and the area that you're interested in. Or some drug discovery targeting Asian-specific disease mechanisms. And I think this is just a general outlay. We will go through some of the discussions. The Pigeonhole link will be live, and we will flash it up again. So you can also regularly follow what people are discussing there. And the end will be a table like this. For each of the tables, we have 5 main tables, but there are more to be filled, where we get the grand chess opportunities, the role of AI. Here, we also wanted to put in what is the data that is required for it, the type of AI, and of course, the potential impact. So going right into the AI and drug design or drug discovery or drug development area, this is an interesting picture because it shows, this is 2022, but it's not very different now, how crowded the area is globally. There are more than 700 AI companies in that field. And most of them are focusing, for example, on small molecules targeting protein in a classical way. So an important aspect for Singapore is, how can we find our niche? How can we be sure that Singapore is the place that is the best place to do this kind of research? And the questions that you can come up with and the areas that you can identify could be those that become niches. So we have some examples where then instead of protein targets, you go to RNA targets. And there are many other opportunities here to make a differentiating factor. It's also about being maybe more Asian-centric in a way, and the clinical data that we have combined for the drug discovery efforts. Typically, AI can be used in the drug discovery pipeline to narrow down the funnel. I think you can't see really the background of it. So it's very difficult for online people to see. Let's say the classical funnel would be like this. It would be wider. So that means in the process from the target to the hit to the clinical trial, you'll narrow down candidates by doing experiments and additional tests. But what AI does, it narrows this funnel. So you can test many more things fast, narrow down to lesser candidates, then go down through. And here on this side, you see a lot of drugs that have come through from more AI-enabled discovery programs are indeed faster. Time spent to clinical trial is less. It's not always the case, and it's some years that it makes it faster. However, the same drugs that are coming from AI-designed drugs have the same problem as the other drugs. Many of them fall short in the clinics. In fact, only 1 of 10 of the drugs makes it through to the final. And this is the, I need to go to the slide again. Yes. This is the whole path for, let's say, the pipeline for drug discovery. You identify targets, you validate it, all of this already can take a long time. Then you identify first hit. Could be, you know, your lead molecule that is being generated, then optimized, then goes into preclinical and clinical trials. So the first funnel that I showed to you, typical applications are here in this area. And but the biggest problem currently is still when you say only 1 in 10 go through, that the cost of failure at this late stage is very high. So this is easily $1 billion if you started a clinical trial and you don't go further. So I think there are a lot of opportunities also where AI can help here. Maybe going beyond the molecular targeting aspect of the drug, but also into the cellular studies, the omics world of data that we have available. So because of this, we have predefined 3 tables. One that is towards finding the right targets, one towards finding the right drugs, and one towards finding the right patients. And in addition, we'll take more of the topics. You know, synthetic biology is one of the ones coming too, and also additional questions that come in. So I now want to hand over and introduce Yaron. Thank you very much for coming. He's our representative for EDDC, our national platform. He will tell us all about it. He has had many different roles already. And maybe I'll let you introduce yourself in more detail. I think I first met you as Human Longevity, then Engine Bio, and now also EDDC. Thanks.
Thank you. First, thank you, Sebastian, for organizing this great workshop and for the invitation. It's a pleasure being here and meeting you all. So I'm focusing on EDDC, which we maybe relate to the previous referral by Kedar to the applied AI research. So how do we translate the information, the predictive models into actual drug discovery? And let me start in terms, there are no slides. I want all your attention to me. So basically, let's just share with you some of my background. So I've been in the pharmaceutical industry at Eli Lilly, AstraZeneca, led the global analytics teams that focus on all stages of the pipeline, from preclinical to advanced clinical trial systems. So kind of exploratory genomic studies to highly regulated systems. And then in the genomic industry was in the early days at Affymetrix, when there was a lot of skepticism that genomics can actually be measured in a high throughput. And then at Human Longevity, when we try to do it at really large scale and translate it to biomarker and target discovery. And when we use the term AI in our context, all it is machine learning based predictive models. What we try to do is simply generate hypotheses, right? So these are in silico computational hypotheses. And what would be the bottleneck after generating this hypothesis? To validate and build confidence in this. So I think as a computational team, which many of us here are, we can generate many ideas and many computational hypotheses. The key is when we talk about the promise of acceleration and reduce failure rate, it really narrows down. Can we, thanks to machine learning, generate hypotheses that we have much higher confidence in them to hand over for our wet lab scientists to validate, and in a level of confidence that basically will reduce failure rate, right? And so the ultimate test of our advanced algorithm and integration of information is really, can we generate this confidence prior to it going to the major spend, whether it's in vitro, in vivo, eventually clinical trials. So time will tell if we can actually deliver on this promise. Now, when we want to generate hypotheses, we mentioned one bottleneck is the validation. But as we all know, and data was mentioned here already 10 times probably in the previous talks, we focus on EDDC on the data foundation. So automation, standardization, I think the used harmonization was used earlier, and the aspect of QC of this information. So how can we integrate information across different functions in a way that minimize human intervention? How many of us are doing Excel and copy paste and take the CSV file as an input to our predictive model? This is not scalable. We cannot have an AI platform that is a one-off. Oh, just send me the file and I'll feed it into the predictive model. And in machine learning, a keyword is learning. So we have to make sure that our machine learning platform is designed in a scalable way to receive data constantly, to serve as validation and the cycle of learning for which hypothesis we take. So we focus on integration, standardization, right? And aspect of automation across different functions to bring the data layer, the data foundation. Once we have confidence in this data, then there will be exponential growth of predictive models that we can trust. And we can extrapolate from this concept of what we do now in EDDC to enable the machine learning. We mostly focus on target, biomarker discovery, small molecule, large molecule. There's aspect of RNA across mainly in oncology and I&I, in immune and inflammation. These are the key therapeutic areas for us. We do touch other therapeutic areas, but these are the core ones. So we need to be measured on each stage or by the quality we'll deliver. But if we extrapolate this concept to the Singapore ecosystem, how good are we in integrating this information that we have amazing, like a gold mine of data sets within Singapore, but how well are they integrated? How many manual steps we each need in order to actually access a portion of this data? And we all went through this pain, right? So sometimes we get access to the omics, but not the clinical metadata. Each one has its own page, if you will. So we have to break these silos at the Singapore national level and connect the data to enable the foundational research, the AI for science, and eventually the applied in order to tap on this really valuable information. Now, if you think about it of differentiation, how many countries out there can be in a position to actually connect such data set, because the data quality is very high in Singapore. So we can really differentiate Singapore by stepping into this integration and therefore enable novel discoveries that can be validated. And remember the loop. Once it's validated, how do we design our platform to accept, to receive validation data to continue and optimize the predictive model and hypothesis? And I think it's not as exciting as developing the next amazing neural network, right? But we should not focus on developing neural network before we have confidence in the data. And the last 30 seconds, maybe if I have, I will just touch another topic, which is the value of noisy data for machine learning. So I know we are pretty obsessed with cleaning the data, clean the data, clean the data, but do we really have to? We need, as long as we automate, we standardize terminology, metadata, ontologies, and we focus on the QC in general, it's okay to have noisy data. And maybe the one example why noisy data is important, because machine learning trying to differentiate signal to noise. We try to detect a signal. And if we don't have the noise, the differentiation will be even harder. But if you take, for example, single cell data, which we're all trying now to get into this very high resolution of information. One algorithm that we deploy machine learning is the segmentation, is the differentiation of what are the different cell types that we have, genomics expression or other expression, right? So we are quite obsessed with separating the cell, identify each cluster of cell type. But what we learn that when you have a mix of cell types, even if you don't separate them, there is a value of understanding the neighboring cell types for the pathway and the biological implication of this cloud of cells, even if they are not fully separated. So we can actually tap on the noise or the less separated cells in order to learn and conclude different types of pathways that are important for the mechanism of action and target discovery. So I think we should value noise data, embrace it, and then feed it into the different features, the engineers, what the feature we engineer into the machine learning predictive model. And with that, I'll trigger this for further discussion on the different discussion groups and look on those different challenges. How do we tackle them at the institute level and the national level? Yes.
I'm just curious, thanks for that. I'm just curious because for example, in materials, we talk about high fidelity and low fidelity, right? Which is no matter how much you do automation integration, the experimental data that you can collect will never reflect fully the complexity of your final high fidelity data, right? So for you to do scale up, for example, that's really hard. So here, even if you spend an effort doing integration, automation, is the complexity of what happens in the clinical trial actually captured? Because then you have a gap, right? Because no matter how well you have a forward model that predicts based on the data you collected, it'll never reflect the complexity of what happens in the clinical trial.
So I think that speaks to the importance of bringing basically translational science. So bring data back from the bed to the bench, right? So it's never in isolation. You don't stop with the in vitro predictive and then just hope for the best. I think we have to have the end-to-end platform to enable the data ingestion from every stage, including clinical trial. And this feedback of survival and drug response, which is always hard data sets to have access to, must feed back to day one, to the early predictive model and see if it applied for the different animal models. But yeah, it's a gap for us, the different organisms and versus human. The key is to think end-to-end on the cycle of the data. That's the way I look at it. Other questions? Okay, thank you.
Thanks, Yaron. We move on. So you saw a very good introduction. Also, what are some of the opportunities and challenges, and how important that the data is? There are many different aspects to it, and our next speaker, I don't know if he will mention that, but I always like to say that. So when 20 years ago I was in Austria doing work protein structure prediction, I was also closely following these competitions on how can you predict a structure from the protein sequence well. And Yang, in fact, has been dominating this competition for many years. And when we all say about the excitement of AlphaFold, this was a very, very nice and good effort that won 1 year, but then lost out again against certain other groups. And the new development of let's say AI for the structure prediction shows also how fast the field is moving, and the very adaptive approach to it is also what we all should take. And I'm grateful, Yang, also for you to tell us a bit about that.
Thank you. Yes, thank you. First of all, thank you for organize this very nice meeting, Sebastian, and also for the invitation for me to introduce the field of the AI-based protein structure prediction. And so when we talk about the protein structure prediction, we have to talk about a very important game or competition, which Sebastian mentioned, is called CASP, C-A-S-P. It's for the it's called the Olympic game in protein structure prediction. So basically, this is like a blind test of the prediction, and this is also the first high-profile event, which is introduce a competition into scientific community. So basically, every 2 years, organizer collect a bunch of the sequence, about 100 protein sequences. Nobody knows the structure. Then they send the sequence to the predictor to solve to predict the structure. In the meantime, they will send this to the structure biologist to solve the structure. And then 3 months later, they will compare your prediction with the experimental structure. So that is really probably the only way to assess objectively what is the progress and what's the accuracy of the modeling. So yeah, long story short, this is what I have seen in the last 10 years about how the very impressive progress of this field, especially made by the AI, by I will just highlight this. So this Cas11 is like 10 years ago. That is without AI introduced. So then we can only fold a very small protein like a alpha helix protein. This is the red are experiments, blue are predicted structure. You can see when 2 years later, then people introduce co-evolution. This, of course, this is not AI, but it's also because of the data increasing and also new technology introduced, make a very good progress. So AI introduced, into the CASP is mainly in the CASP 13. You can see the folding rate is increased dramatically because of CASP 13. And we are able to fold larger protein. So the most significant one is in the CASP 14. And we are, okay. So this is the CASP 15. So why the most significant is CASP 14 is because we are talking about is AlphaFold 2. Actually, AlphaFold, DeepMind participated in the CASP 13, but they are not really stand out this significant. So AlphaFold 2 in CASP 14 probably, to me, I think is the most important revolution in the history of protein structure prediction. So what they did is that they developed like a so-called end-to-end neural network, and using a very cutting-edge AI approach is called transformer. And then they specially introduced a new module is for this like a structure module, convert the 3-dimensional structure into this into the network, so that we are able, the first time enable from a sequence to learn the final structure. So you can see those are really amazing prediction. And this again, this blue is the experiments, and green is the predicted structure. It's almost identical to the experiments. Those are blind tests. So one question, actually, many people ask me, why AI is so powerful? If we show this data, those are, I can say 3 classical methods, like charm. Well, this is a charm, so I spell it incorrect. That's a molecular dynamic simulation. It's mainly based on physical force field. They have, in general, 7 energy terms, about 20 parameters. So they are not really can fold a protein, basically. So they are mainly used for structure refinement in the structure field, or you can try to see that trends of the protein folding. So this is iTensor is based on energy, based on the knowledge-based statistics from the PDB library. Then we have 26 statistical energy term, about 100 parameters. But if you look at AlphaFold 2, they are having about 10 billion of the parameters, which you can learn, from the PDB library using this neural network. So apparently, the limited number of the parameters, so it's limited the power or accuracy of the statistical potential. But using AI, this I think to me, I think is critical. You have so many different parameters and so big structure data set, then that will enable you to really go beyond the traditional statistical method. So that is, as I mentioned, is Catapult 14. So 2 years later, is make a lot of progress. This is in Catapult 15, and you can see now the AlphaFold 2 is here, and there's many other methods doing better than AlphaFold 2. Of course, slightly better, and of course, there's many others is sticking on the older method that not is actually much worse than AlphaFold 2. That's for single domain. But if you look at the multiple domain or protein-protein interaction, many other methods is really doing much better than AlphaFold 2, because AlphaFold 2 is not really trained for this multiple domain or protein interaction. And so here is a very interesting slide, because this shows that, previously we are doing the protein interaction is mainly based on protein docking. Now, when we introduce the AI into the protein-protein complex, we are able to form a very big protein-protein interactions, like some of them more than 10 chains, more than 8,000 amino acid. So, but if you look at this comparison, the black one is the single chain, and the red one are multiple chain. So although the AI make a very big progress of protein-protein complex, but still their accuracy is much worse than the single chain. So that highlights the fact is that in this field, PPI protein-protein complex really remain as a very challenging problem. So that is also a problem we want to highlight in this field. So this is probably the last. So this is what we have done most recently. We try to apply 2 state-of-the-art methods, AlphaFold 2 and the DI-Taser. DI-Taser is a new version of I-Taser, introduce the AI into the traditional I-Taser. So we apply this to human genome structure modeling. And the data is not yet published, but I just show you how, because one of our goal for development methods, we really try to fold all this protein in the human genome or in nature. So here, that is the full chain in including multiple domain. Those are what DI-Taser is doing. So those are the genome which is in different chromosomes. And in this app, we have about 20,000 protein sequences. So when we do that model, we found that there is about 1,000 protein has been solved by experiments. So we compare, that is after our data released. So we compare those one which have the structure of this DI-Taser and the AlphaFold. So they are very close, so they are complementary. So this is what we have seen of this DI-Taser and the AlphaFold. You can see this in this region, both methods can fold all these proteins. And this region, which DI-Taser can do, but AlphaFold cannot do. This region is AlphaFold can do, but DI-Taser cannot do. So we can see there is still a substantial amount of this protein, which neither of DI-Taser or AlphaFold can fold. So that means, although the field make a really big progress, but there is still a substantial amount of protein which current method cannot work. So that is one of the reasons is that for those ones are orphan proteins, they don't have really homologous in our data set. We cannot derive the co-evolution. So without the co-evolution, deep learning cannot train or predict well. So those are one of the major problems. Also, there's some of the protein, there is 4 of this, this disordered region. So that's highlight the 2 problems which I want to highlight in this last slide. So the progress is that we are really very close to nearly fold all this single domain protein structure prediction. So that means we are probably solve the structure for the problem to the single domain protein. Second, for more than 50 years, this field is dominated by homology-based modeling. So basically, if you want to predict whether you can fold a protein, you need to look at whether you are lucky enough, have some homologs from PDB. So now, we don't really rely on PDB. We are more relying on the genome sequence. So there's a paradigm shift from relying on the PDB structure library to on the genome sequence, because genome sequence can help us to identify the co-evolution. Then we can learn using AI to learn the predicted model. But there's still some challenges. So that is also what Sebastian and wanted me to highlight is that what is the challenge to the field? To me, I think it's sure that the data significant difficulty is still remaining on this multiple domain protein and also protein complex. So we have much less confidence about this compared with the single domain proteins. Second, based on AI, we are not able to modeling how protein fold or what is the protein dynamics of folding pathway, including this disordered protein. Also, I just mentioned another very difficult problem is that, for the orphan protein, which we don't have homologous in the sequence or in the structure database, how we fold those of the orphan proteins is still challenging for either single domain or single chain and a multiple chain protein. So the last one is that we're making bigger progress about this, but still a problem is that, the metagenome is become huge and larger and larger. So, but our computing resource is still limited. So nowadays, if we want to really scan using traditional method to scan the entire metagenome, we probably need many days. So we try to shrink the database using some skill, which we can marginally do that in about 1 hour. But there, the genome sequence is still increasing really exponentially. I think this can become more and more a challenge, how we can quickly, accurately derive the multiple sequence alignment and then to derive the co-evolution. So this field is mainly, put a lot of effort to train the model, but this is a very important problem, but I think it's overlooked by this field, but I want to emphasize it here. So we still have some of the opportunity, We are, as I mentioned here, the AI is a black box. They don't tell us how protein fold. So I think one way is that we combine the deep learning with the physical-based folding simulation, then we are probably able to derive some dynamic signal and also model like an ensemble of the conformation. So another way to deal with the other problems is that we try to combine AI with this cryo-EM. This is a very powerful structural biology tool, but in many cases, cryo-EM, the resolution is not enough, or some of the data is sparse. So combine this cutting-edge structural modeling tool with the cryo-EM. So that is really give us a very powerful, promising. So yeah, I think this is all for my talk. Thank you.
Thank you. So the format is that we go very quickly through the presentation, but if you have one quick question, we can take it. Yes, Yuming?
Hi, yeah, from the perspective of computer science, do you think the protein sequence contains all the critical information that we can predict in the protein structure?
For the sequence, of course, in nature, all the structure information is encoded or embedded in this protein sequence. But we cannot predict. Actually, now we don't have any method, just predict from this single sequence. we needed to derive the homologous co-evolution information. That is derived from the multiple sequence alignment from many homologous. Because that homologous, basically, we can derive how the evolution comes to this, because evolution is very important for the function of the protein and then decide the structure. So that is a key point of why AI can help us to fold the protein.
So a further question is that can we predict the protein structure by genomes?
That is what AI is doing. So if you have a really a large, let's suppose we have a complete genome, and then we can have multiple sequence alignment, very deep multiple sequence alignment for all this your target sequence. Yes, the AI can fold all these cases.
Sounds good. Thank you.
Yeah.
But just one more question related to this. Is it one-to-one mapping? For example, given one sequence, is it possible to have multiple different type of structures due to other factors?
Yes, that is a good question. And, actually, many of the proteins, that's the reason I just raised that issue about the dynamics. Actually, in protein in the cell is not a single static structure. They are fluctuate. Some cases, they probably have multiple state, or depending on your cofactor or how they are, interact or binding with other binding partner. So that is what AI cannot do, because AI is trained on the data on this stable structure, solved structure. So that is exactly the question which I think is challenging to this field.
And in fact, AlphaFold and other methods where they are not sure how good they predict, this is the part that is most flexible in the protein most often. Thanks, Yang. So we move on quickly to Hao. Hao is from BI, but also a chunk in NUS and UCAN-US.
Thanks, Sebastian, for the introduction, and thanks for the nice opportunity. So I'm a senior PI in the BI STAR. My lab focuses on a few things. One of them is some molecule ligand discovery. I wouldn't call drug discovery because none of my ligands have got to clinical trial yet. So do this. As Sebastian already mentioned, this drug discovery pipeline. So you can see drugs include, some molecule to peptide, to antibody, to nucleotides, even cell therapies. One common thing among these therapies, they all take a long time, 12 to 15 years to develop. So that's why we want to use AI to speed it up and make it cheaper. So my lab, one focus of my lab is ligand discovery. So some compounds we have developed on different systems have already got to preclinical stage. So for example, we have working with our NUS collaborators on this human virus on the left side. To find repurpose a drug, market drug to target human virus. This quite successful. Will be published soon. So we're also working on NCCS on a kinase target, and also with the National Neuroscience Institute on it's a neurodegenerative disease, Alzheimer's disease. We also for both target we have filed a TD recently. Yeah, we will file patent soon. With the NUS collaborator, we also target an ion channel. We have developed peptide therapeutics, which also already been patented. Also together with our GIS collaborator, Ram, who is sitting here, we develop a small molecule inhibitor, protein interface disruptor between beta-catenin and TCF, which is in the middle. Yeah, this just heard from. We have patent and publish it. I just heard from Ram. It looks quite promising, going towards the clinical trial. That's very happy to hear. So, so the past 10 years, we have some experience in this field. So one experience I learned is for each of the work takes 3 to 5 years, just only for the political stage. So that's we want to make this faster, and we want to apply this to many more targets for most proteins in human genomes. So along the drug discovery pipeline, there are different stages: target validation, hit identification, little optimization, and then preclinical study, clinical trial studies. In my opinion, the hit identification is one of the bottleneck because, as I said in the previous slides, it often takes a long time to get a hit. And many proteins in human genome hadn't been targeted or hadn't have any good hit identified. So when I joined BI 10 years ago, I give a presentation. Maybe Sebastian still remember the figure on the top left. I plan to find small molecule ligands, the hit for all the human proteins on all their binding sites. So of course, this is a big dream, but we want to. We have been trying to work on it. The first step is to find a hit for this target. This is our aim here. So with the support from NIH as genetic grant, we get, 4 years ago, a very talented computer science student, a few PIs in BI working together, me and other people. Yeah, we develop a method. It's a machine learning docking integrated method. We just recently filed a patent for them. Here, don't take a serious of this figure. I just take them from a paper. But they basically tell us what we do is we train on many proteins on their ligands and also some assumed decoys. We also put a lot of feature into the model and then train different machine learning model to see which one better, and then modify some of them, make it better. So this method has been patented. It's working pretty well. When we test this method on external target, benchmark stat on the left, respectively, on 22 target, which is far away sequentially from the training set protein, and the chemical ligand of them is also far away from the ligands from training set. We get a pretty good hit rate. So among on the histogram here, so on each of the protein, the top 10 ranked compounds, then we check how many true ligands are there. So you can see for some cases, we get 100% accuracy. Some of the lowest we have is 30% accuracy, sorry, 20% accuracy. Yeah. So this is a near perfect performance here for this external retrospective validation. The good thing is the method is about 10,000 times faster than other state-of-art approach. That means we can apply this quickly to almost all the human genomes, screen millions of billions of chemicals in a day. Then following work is just pick them, give our collaborator to test if we can find collaborators. Yeah. So of course this is just retrospective study, playing with benchmarking, playing with the data set. You can people can get good results. Okay. So the real thing is you test on a real scenario, prospective validation. So through my collaboration, I ask people because I don't have money to give them, I have to do them by their own money. So I have a few good friends over the world. We pick 5 different targets, different from the training set. One of them I can tell you, I'm showing here on the bottom, is have almost no hit available. But basically, we cannot learn from existing knowledge. So each protein takes 5 minutes to come up with a prediction. We did some docking to the final good scores compound. Yeah. Then we give 20 compounds for each target for a collaborator test. So we are happy that for each target we tested, the hits will come out of this 20 compound. So we are doing further studies on them. Yeah. Hopefully solving some of them, some actually structure complex. So this is I can show you. So we get I can tell you the hit rate is 10 to 20%. This is not satisfying, in my opinion, because we from our retrospective study, we get a 20 to 100% hit rate in top 10. So here is something like a 10-20%. This is we can still do in my lab if we do just the traditional auto screening. We get still 10-20% hit rate. That means I pick 10 compounds for screening, one of them will be good hit or 2. So that's accuracy is not significantly improved, but speed is 10,000 times faster. The next thing we'll do is to improve accuracy further. But we know a few things to try. Yeah. So this is a hit identification. Yeah. Then the next step will be the lead or hit optimization. So we need to explore the chemical space from the existing hit, which in my opinion is not the most important, is not the most hardest thing. But compared to hit identification, which you need to screen out of 1 billion compounds, here you need to screen a few thousand. So you optimize, you search around neighborhood, basically make some chemistry knowledge with MD simulation, FEP, financial calculation. You can get within a month of time, you can get some good guess of the good analogs, better analogs. So AI can help, but I wouldn't say make it 10,000 times faster or better. Yeah, that's not possible. So, but still, there's space to work on. So far, I think people have been trying to including molecular properties into this detailed web validation, means sensitivity, cell toxicity, and membrane permeability, but hasn't been widely applied. One recent work I want to highlight by the called PigNet, which I like the most, is they do this binding affinity prediction. They use a graphic neural network to generate all the features, fit velocity model. Then they try to predict different physics terms, van der Waals, hadron bonding, hydrophobic, those terms. Then use this to do binding affinity prediction. I think this is a nice way. Maybe we can replacing this classical mechanic terms by quantum mechanic terms to make them more accurate. There are different things to do it. This is one thing I would say in later this future challenge. Yeah, but one thing we can play with. Yeah, but here compared to heat optimization, it's a heat amplification. It's a less a bottleneck, but still a lot of things can be done. Following this chemical space exploration, I would actually introducing the chemical space generation using AI, which a lot of work has been done. I think it's going quite well. We can still also pushing it to generating novel, non-existing chemicals using AI. There are a few methods already available called RN, the GNN, and also VAEs. So people can use this to generating new chemicals. Also, in the same time, considering their physical, chemical, even drug properties. Then will save a lot of time for later. People don't need to fill the compound because they are too toxic, cell toxic, and not membrane permeable, this kind of things. So this is feel very promising. Yeah. And the last slide, second last slide would be the AI clinical studies. In the past 10 years, there are multiple models predicting ADMET, PBPK, and also clinical trial outcomes. But most of them are simple AI models, like, for example, this tree-based approach, shallow models, yeah, regression models. But recently, I noticed some work called this ADMT AI and this HINT, HINT. They start to use graphic neural network to combine different resources from drug, from patients, from disease. This can help us bridge this hit to lead to later the clinical outcomes. So we feel this is AI can also more work can be done here. So finally, let me just summarize. I want to mention the challenge for binding prediction, including hit identification and lead optimization. First of all, is people already mentioned the data quality. Yeah, because many experimental data we can use to train have been generated by different labs for the same protein, which is not necessarily comparable. Yeah, so also we don't have a lot of data on the negative data. Yeah, maybe it's hard to we have to generate some synthetic data or something like that. That's how the best way to do it is an issue, and the accuracy, as I showed you, still need to be improved. And most time AI will rely on the data, so it get a bias from known knowledge. Yeah, so how can make an AI model more transferable, but generalize better? So one thing I'm thinking is to have this AI model somehow fit into, including integrated with physics and chemical rules. Yeah. So I wouldn't mention too much on this generative model on chemical generation because I haven't started much things there. But I think for the preclinical and clinical studies, apparently there are challenges is data scarcity and their bias based on human population. Yeah, I mean how the data are shared, different countries, different continents, different ethnic groups. So then to make this lead to the problem of generalizability and validation. How can the model be tested on different populations? And finally, because this part bridge different gap from different type of data, from binding to cell accuracy to later animal model to all the patient response. So how can we make an AI model more interpretable and explainable? So when things go wrong, we know what is wrong. We can improve it. So this is something I think is a challenge. But the good thing is now in Singapore, we have this SG 100K. In A-Star, BI, also other RIs, we have this national cohort data. BI hosts Biometa. So we can try to accumulate more and more data and have an initial model, AI model, and gradually improve it with more and more data. It can be shared more, openly. Yeah, that's what I think. Yeah, thanks.
Thank you very much. One quick question. So you can put your question also in the chat, but you can ask one now.
Lead optimizer.
Yeah, lead panel. Sorry, thank you. Did you perhaps try to also use your validated, data that you send for in vivo and in vitro testing and sort of use it back into your optimization steps to see if some of the you see a spectrum of like performance, right?
The model I showed from my group is only the heat application. It's not lead optimization. We haven't developed that yet. Yeah, but it shouldn't be hard to develop. There are a lot of data, strong, mediocre, weak binding with some data. The quality is an issue, but you can still use, especially apply for specific target. There shouldn't be a problem. I mean, we should have a good chance to have a good model which can predict the correlation. Yeah, but just now you are saying whether we can use that to feedback. Yeah, depends on the amount of data you have. If you have a few thousand data points and you get a few more new validated data, well, okay, maybe 10 or 20s because include negative data, it will help, but it wouldn't change things significantly. I mean, if it's small amount, it's just from one testing. But if you test many, it will definitely. Of course, we should always get things feedback and yeah, definitely. That's the case. That's the case. I agree. Yeah.
Thank you, Hao. So the selection of the speakers also was to somehow fit together. So Yang was showing you how can you predict the 3D structure. Hao was showing you how can you find a small molecule that binds to that protein structure. And now we have Yuguang from NTU who will come with the same task to bind a small molecule to a protein, but try to do it without structure. So it's a very interesting new approach, and I'm happy to hear it.
Thanks, Sebastian. So here I briefly introduce our recently the work, which as Yang mentioned, right? Now the AlphaFold 2 or his model already from only sequence level can predict very accurate structure model. That means the structure information already hint in the sequence. So now can we do that? Can we do that structure free? That means based on the only sequence information, protein sequence, and the small molecule SMILE code, which also can we do this prediction directly without directly resort to the structure information. Here we come up a so-called the bind model. And this model, I do not go to the architecture, mainly is the cross attention between the protein large language model, ESM2, and the graph model from small molecules. In fact, this idea is not completely new. 2023, one paper in the BIB, which is done by the Chinese Microsoft group. They call the SSM DTA, which also have the same idea, which is without direct go to structure, then see how the binding affinity prediction only based on sequence level, how the performance. Here we do here they are also using a cross attention mechanism and do the prediction build up a model. So okay, I just saw a few slides. What's the performance? So in the virtual screening area, right? We have a lot of well-established benchmark, which we call it the screening the EF enhanced factor, which is there's no answer. Then we just do the blind docking or scoring or ranking and see how the hidden target can be identified. So usually the number bigger, that means your method have much more the enrichment factor, so that perform better. So you can see from this CAS 2013 the data set, there is total 285 protein ligand pair. Then we can do the study and our model band, right? It's comparable to yeah, I think it's a little worse than the best structure-based model. And also compared to SMDTA, we are quite good than that. This is one result. Another result from this Disco 2.0 point, this dataset, which have 8-1 new structure and the protein sequence. So we are in fact band, right? The performance is the best. All other model, right? Nearly besides the SSM, DTA, all other model are structure based. Also from the, I think in all area, right, they are very well known. We call the DUD database, which has 102 protein, and each protein have quite a lot of active compound, as well as the manually generated decoy, which is the true false binder, and to see whether each of model you can distinguish between true binder from the whole much more decoy molecules. And also in this task, right, our model is perform quite good, and also SMDTA also not bad. Yeah. So here another very recent way, another data set which has contains 15 proteins and 8,000 true binder, and also more than 100 times, or 1, 2, 3, yeah, much more the decoy, which is the false binder. This exercise, right, is very close to the real, we call the virtual screening campaign, which we virtual screening large small molecule database and only choose a few, we predictable true ligand, and they found that our model also perform quite well. Okay, compare also the SMDTA also is not, yeah, is also quite reasonable. So in other words, all this exercise, right, to show that all sequence-based model can really comparable, or I cannot say very much better than that, the structure-based. So we do not, all the exercise, right, we only input the protein structure, protein sequence and the ligand sequence. So another, we also do the reverse docking. Reverse docking means that usually in the experiment, right, on the phenotype experiment, they found some good ligand, but they do not know which protein in the human they bind. So this is the target identification. So we do this exercise. We choose from the 90 protein ligand pair, which is known in the PDB data bank. Then we take each ligand, then we search all the human genome, which is from the AlphaFold2, the genome sequence. Okay. Of course, other model, which is structure-based, they take the genome structure from AlphaFold2. Also, of course, younger model even better. But here we take AlphaFold2. Then we see if any of the model, right, can select out the true protein. And our model, in fact, is perform the best. So about top 100 means that you each series, right, talk from the 12,000 protein human protein, we choose top 10 candidate, and this top 10, top 10 or top 100, then the true protein target is within the top 100, and then we found about here at this point top 100, we found nearly 60, more than 60 out of 90 protein we can find. So another database is called Aztec, which recently suggests the reverse docking exercise. There's total 85 complex. So we performance also quite nice. Also, we recently, in fact, we also come with a startup, and we summarize all with this bind, right? We can, really can virtual screening a large data set, but we use the bind as a first filter. Whenever you have a good drug target, for example, usually for this method, right, only protein, then we quickly screen filter. Then we can use the structure-based, for example, Flex or Bina plus our own SFCT correction, which also machine learning-based score function. Then we do finally, we usually do the evidence submission to validate, because all the docking or somehow ignore the dynamic picture. Then we purchase the compound, then we do further the wet lab validation. So this is all pipeline. In fact, right, our recent work, I don't mention here, but we also can study not only protein, but also RNA. RNA structure, which is structure based. Then we we choose nearly 3 million library, then find a 10 compound. Then we do the cell-based experiment and found that one or two compound have low IC50, I think around the micro or even less than 1 micromolar to kill for example, the pancreas and the lung cancer cell line. So it's quite nice, which greatly expand or target not only on protein but also on the RNA side. So yes, I think here I finally come up some question is that, for example, this bind, right? Although it's nice, but here we can see we do not find very good the explainable, because all the cross-correlation transformer is quite a black box. And another question that we already have, for example, PDB bind date, already have very clear the structure information. Can we use this information to go to the training, then come up a better model? I think this is the one difficult task. Another combining or own the AI, right? For example, I also recently applied for the SG100K, which we can available multiple model and multiple omics data. Then together, can we come up, from the drug discovery, can we go up a little upstream, which we can find a novel drug target for certain disease or something like that. I think this we can think about that. Okay, with this, thank you very much for your attention.
Thank you, Yuguang. In the interest of time, we will move on quickly, but of course it will be included further in the table discussions. I think it's very encouraging results, and the power of protein language models, I think, will also be featured again in the synthetic biology part. We move on now to
one last question.
Yes.
Our model is now online, is fully open access, so you can search, then you can download. Yeah, try.
Thanks. It's also good to share the models and try it on different data sets. This makes it also a step towards being more explainable. So Mila is gonna give you a part that also Yuguang mentioned at the end. Instead of protein as a target, maybe RNA as a target, and the RNA structure prediction. Yang's lab also is doing RNA structure prediction. Already has a method out there, and we will have Mila describing a very interesting local effort how we are contributing here.
Okay. Thank you, Sebastian. So how AI is so important, I put too many AIs in the titles, sorry. So yeah, I will talk maybe about something different about generative biology, but I will start with some goal we set up 3 years ago, how to make RNA druggable. So as Yang mentioned, CASP is important for AlphaFold. And it is interesting in the case of RNA structure prediction on the last CASP, AI methods underperformed. The best methods were still thermodynamic methods with some human intervention. So with RNA structure prediction, we have many, many more problems than with proteins, because we have much less structures that they are determined, and yeah, and also there are much less people in the field. But on the other case, this is good. So here it is some kind of idea how we can construct the RNA structure prediction, but I would just focus on 3 components. The first one is that we don't have enough structures. So it is important to try to score some structures from sequences that do not have resolved structure, so that we can use them in some kind of self-distillation. So, and for this, it is important how we will score if the structure is good enough. Some people in the field can tell which structure is better or worse, but can we learn the models? And it is interesting work we do collaborate with CIFAR, with Eva and Yanga, is to try to improve the current methods as RS to provide better score. The second component is can we add some experimental data? Can we add some high throughput structural probing data, like shape, splash, or different data, try to improve our models? And the third one, it is, from my perspective, was the most interesting. What is the input? So as Yang mentioned, it is important to have co-evolutionary, evolutionary information, but multiple sequence alignment for RNA, it's not so good as for protein. And maybe because of the lack of people in the field. So at the beginning, we decided, okay, what we can do with RNA language models. So we recently developed the first version of our RNA language model. We call it Renaumo, and at the moment, it is probably the best and the biggest RNA language model. So we didn't do so many changes comparison with some other language models like RNA-FM. But what we did using our industry experience, we train it properly. So how to train the model, even if you have all components, how to log, how to not spend too much money, it's also an art. So the current results, we achieved that even people believed that we don't need bigger model, we show that the bigger models are better. So we trained initial model on 30 million sequences, and now we are training the bigger one, and we will have to use 1 billion sequences. As also Yang mentioned, there are a lot of metagenomic data, but it's very important to be careful about this data because it is not sure about their quality. But now we are using this data to try to learn the bigger model. And also we try to use some shape data in some multiple ways. So how to add additional data in train the model. So we did this in collaboration with Hwee and Roland. So this is some kind of results on downstream task. So once when you have the model, you can use it for various downstream purposes. Here, I will just briefly show you the results on secondary structure prediction. It is important because people before thought that using deep learning, you cannot generalize well for secondary structure prediction. So for example, when you train on one family of structures and then try to predict on other, results underperform. Oh, sorry. So results, you can see on the right, there are 3 deep learning methods underperformed, or results on the left that are first 2 columns are 2 state-of-the-art thermodynamic models. But when you carefully train a language model and then using the same families, so always leave one out and use the Renaumo and some additional fine-tuning, and we use the ResNet, we achieved on all except one much better results than with thermodynamic models. But it is interesting that we achieved the worst results for one family that actually deep learning models work better than traditional methods. So we are inspecting what's going on, but we think we know that we can improve. So we show that if we have enough evolutionary information, then using some fine-tuning and even on families with unknown secondary structures, we can outperform existing models, and this gives us many confidence for our future work. So what are the opportunities? Of course, there are many new AI, foundation models, diffusion models, JEPA. I really like JEPA, and we will try to see how to implement it. In the future, we will need to integration of protein and RNA models. There are lots of discussion and how long can be RNAs that can be alone for that they need proteins or something like that. Some people say that everything about 200, it is not easy to predict. Also, I think there are interesting work that Ron does. It is antibody prediction, but it is much related to proteins. But there are many challenges. So the first one, we have seen that now AI is taking over in many fields. So in image, in games, text, and but can we do the same in biology? The main difference is that for all of these, we know how the correct answers should look like, how the sentence should look like, how the image look like. But do we know in biology what is the right answer? So for example, I can use the generative model and produce some mRNA. How I will know the sequence? No, we need experimental biologists. So I really believe that it's important that people work together. And I never liked the paradigm. So we have a lot of data and just try to find something. I much more believe in causal ways so that we can do some perturbation and then measure. And I think this is important for future. And something that we discuss on our workshop is that we really need to train the new generation computer scientists that will start from the beginning of their education working in biology. From my experience, and I'm a computer scientist, I've worked in different fields, from physics, biology, finance, social. And always it takes at least 6 months that people can start talk together with the same language. Sometimes people think just as important, oh, you have good biologists, best wet lab biologists, and you have good computer scientists, and they will produce something. No, they need at least 6 months to start talking together. And so, for example, Roland spent with us almost 1 year talking and trying to explain many things relating to RNA so that we could start working in the field. So, Sebastian asked me about few challenges. So the one, I think it's interesting, how we can make generative RNA language model and that we can have some good prompts. But it is different language. So we cannot input prompt like in ChatGPT, because at the end, the model will learn something from prompts and you will not have ideas. We have some ideas, but I think this is one of challenge. So for many other things, for proteins as well, and there are some solutions, but I've never seen solution for RNA. And finally, I really believe that data is important. Without proper data, we cannot do anything. So, many years ago, many precision medicine projects started with this slide, so that medicine at this moment is imprecise, that they are not. So you can see here how good are some drugs. So the blue ones are the percentage of fraction of people that are really helped with this drug. The red are those that doesn't. For example, this is cholesterol. So I believe that we need first to try to integrate existing data in health records and health promotion board with genetic genomic data, and then we can start investigate what are differences, and then we can try to find a better drug or using existing drugs, but much more precise. So that's all. Thank you.
Thank you very much. We can move on to the next and final talk by Enrico from Duke NUS. In fact, I've seen his slides, so he's a nice summary of from the beginning what Yaron lined out to the challenges listed, and he has another interesting twist that I like. There are ongoing projects already on, let's say, AI quantum compute, and I hand over to Enrico.
Thank you for the invitation, and it has been great to actually have a very detailed and nice introduction about the technologies, about the state of the art, I would say, right? So I work at Duke NUS, and my group primarily is focused on identification of targets for development of drugs for complex diseases. So my focus is really try to find the best target and then take it for identification of a way to modulate the target and then test it in vivo and so on and so forth. Can this advance in some way? Okay. Okay, so I didn't know really how to pitch this, but I started by looking generally what are the main approaches that are used in drug discovery and drug design. And historically, these are probably the two most commonly used approach, structure-based drug design and ligand-based drug design. But actually, there are many other approaches that have emerged, and just by looking some surface literature search, actually, for each of these approaches, there are already very well established, and today we have many of those AI-empowered approaches. So that many of these, or all of them almost, have been very well designed and enhanced through AI. We heard a lot about this today. But that's probably my cornerstone slide, right? Which I found is which is moving. Okay, so I found this sentence in a tech news from drug discovery, which I would like to read to you, right? So the one thing that is a starting point and you cannot change is the target. You can have all the technologies to develop your best molecules, your best ligand protein interaction. If the target is wrong, meaning that it doesn't have an effect or is not validated and it's not coming from human, all of the drug discovery pipeline will effectively fail at the end. And I'm raising this because historically, many drug targets have been identified sometimes by high throughput screening in cells, in animal models, and very few of them have a strong validation or support from human genetics data. So, for instance, in this paper here, published by John Whitaker and in 2000 and friends, right? In 2005, they did the first kind of systematic survey looking at FDA-approved drugs. And they show and they estimate also that if a drug target has a support from large-scale human genetics, let's say GWAS or all these other approaches, the chance for the drug to actually have an effect don't fail in phase 2 clinical trial is double a target without genetic support. So this tells you a lot how important is the identification of the target, because the power of finding the targets in humans rather than in cell lines or in mouse model, I think, in my opinion, is very important. But also, and I'm getting now to the next point, which for me is very important, is data, right? As we mentioned earlier, and it was touched earlier before with different flavors. We have now quite a lot of data, but sometimes we lack some of the important data. For instance, we lack real-world data on high throughput screening, because some of these are proprietary, they're not shared with us, and that will be very useful to train AI models, for instance. We don't, there are a lot of data coming from clinical trials or other kind of record, what is here is named text data, which are rarely accessible, and sometimes they are structured, they're very sparse. I believe having access also to this data will be very important. And for the identification of the target and the consolidation of the target, I also think that using a multi-omic approach, now that we can do it in large population samples, because there are many omics technology that are becoming rather cheap, is actually very important to identify a target with the real biological effect. So for another approach that I'm very fond of is this enable or AI-empowered virtual screening approach that are now replacing the traditional high throughput screening approach. These are coming to life. They came to life not so long ago, but they are becoming very, very powerful. And actually, I'm going to show you one example that we did in my lab with this. But one thing that, again, going back to posing some questions here is whether to run this virtual screening approach, which use different, generative models, for instance, or other kind of methods here, whether we have the right data, both in the terms of how are we going to model, are we going to featureize, I learned this term today, the structure of the molecules, what's the best representation, as well as whether we can have a collection and we can use all the data that we need for the chemical properties of the small molecule that we want to use to train the AI models. And another approach, another important point just mentioned just before me is multidisciplinarity. So I think this is essential and crucial because AI has great promises. We are today, and it does work. But for a drug development, you need to have all the other expertise in the team working together, talking with each other, learning the same vocabulary. Without learning the same vocabulary, people will talk Chinese, others will talk Italians. They're ending nowhere. So for me, this is very important. And to me, the minimum set, let's say, if we define minimum set, has to include, of course, AI scientists, chemists, and biologists. Because if your drug doesn't work, it's not the problem of the AI, maybe. It's the problem of the cellular system we are testing or discovering, or the animal model, or something else. Or it can be the chemist. But you need to have all of this on board. So, and this is just one slide of what we are doing in my lab and how we are planning to develop this. And I hope this can give some suggestion to this audience of where probably the future is going, or what opportunities are, let's say, in Singapore here. So we discover a drug target for fibrosis. This WWP2 gene actually specific as from in humans. Starting point. We don't want to find targets or genes that modulate fibrosis in mice. We want to find genes that can be targeted for fibrosis in humans. And then we validated early on in mice. And after we discovered the gene, of course, we needed to understand the mechanism of action of the gene, of the target in every different system. So we systematically started the fibroblast compartment, the macrophage compartment, then we moved to another organ, and we're doing the same, fibroblast compartment, to understand in which cell type and the mechanism of action of the isoform is more relevant for fibrosis. And then we collaborated with the company to do effectively a virtual screening using this AtomNet approach, which is actually a deep convolutional neural network method on the complex between any potential ligand and the target to screen in silico molecules. It was very, very fast. Then we did a couple of screening also using structural analogs. And I can tell you that the approach in principle works because we can identify molecules that in vitro at least have antifibrotic effect. These are coming purely prediction from the virtual screening. But this is not enough because this is only one chemical series effectively that we're now optimizing and progressing. But by learning a little bit from what people are doing here in Singapore, I came out that we have great expertise in quantum technologies and quantum algorithm. And one of the limitations of some of the AI methods and this generative model is that the ability to generate molecules is limited. The ability to generate molecules with the right property is limited. And this can be potentially implemented or can be boosted if we have some part of the algorithms or of the pipeline, let's say, using quantum computation or hybrid traditional computation. So I went to talk with Jose Ignacio, which is actually here in the audience. And I asked him, why don't we try to see whether we can integrate the expertise in quantum computing and algorithm in this area with expertise in artificial intelligence and expertise in wet lab biology, which has to be part of the equation here, in my opinion. And I'm very thrilled, actually, for this project that has started already, where we are trying now to develop new methods like a quantum neural network or quantum GANs, effectively, as a starting point, because we want to really leverage the power and the expertise here. And again, the emphasis, again, to have a multidisciplinary team, that I think is a winning card, and a multi-center approach, actually, because we can actually put together forces coming from the Center of Computational Biology of Duke and US and the Center for Quantum Technology. So I also drafted some challenges and opportunities that, as Sebastian asked me to, which are summarizing what I was telling you before. And I think one area which is still probably underdeveloped and is try to use AI model to predict early on adverse effect of targeting the particular biological target. This is very important to have this prediction early on before going to preclinical model, because when you have a small molecule, when you have a target and you need to, let's say, scale it up the project, the first question every VC investor will ask you is whether how is your target better than the current drug in the market in terms of side effect and efficacy. Actually, efficacy is probably less important at the first stage, and the side effect, the adverse effects are very important. And there are some AI model now that they claim that they can predict at the beginning the prediction of adverse effect, as well as other important features like the druggability or off-target effect in multiple systems. Then we also talk about using experimental data to feed and retrain all the AI model, right? Maybe experimental data. However, here there is a problem because many of these data are proprietary and we don't have access. They are very scattered. Also, another important thing is, in my view, is to try to take advantage of the large amount of information which is already there, right? Coming from clinical trial, publication, and also use expert domain insights in the early, when designing the best AI architecture for this aim. Oops. I think I should have a final slide. Yeah. So these are probably three major general statements, right, that can be potentially considered here. So I believe that the integration of multi-omics data, especially in from human patients, but not just, is very important to the most robust and reliable identification of the optimal drug target. So I think that in Singapore, we have, for instance, with the Precision National Program, we have this opportunity. We have genome sequence available for free for everyone to study, right, to say. Probably here the focus should be more on disease cohort rather than the general population. Because in my experience, for instance, when we identify the gene, WWP2, the gene is, you can find the gene regulating fibrosis if you study a disease cohort, not if you study a normal cohort. So having access to well phenotype, disease cohort, and the omics data, I believe is very important. Then the other point is how can we retrain and validate the AI approaches using experimental data? I think also here there are a lot of opportunities, and incorporate external knowledge or prior information in designing the AI. So in my view, a model still has to have a lot of information coming from humans. In a way, it has to be designed for a task rather than a kind of a general model that holds great promises, but is not specific for a particular task. And I think I stop here, and let's start a discussion.
Thank you very much for this. Well, so this concludes the first part for the AI in drug design, drug discovery, and maybe we can ask to flash again the pigeonhole code so that the over 100 people online also can contribute and put in your questions or vote for the questions that are there in the live code. We will discuss all of the elements discussed by our speakers and the challenges also here on the tables. But then in the breakout session, we cannot include the online audience. So it's important that if you want to influence also what is being discussed and bring in your own ideas, please do so via the pigeonhole. So we are now moving to the synthetic biology part. So we have 3 more very short talks left. We'll start with synthetic biology with Hwee from ISTA, and we have several efforts ongoing in Singapore. There is Syncity in NUS. Our colleagues Matthew Zhang couldn't come today, but what we're going to do is present a general overview of how AI can contribute. So I'm happy now for Hwee to take the stage and come with a quick summary of some of the thoughts how AI can contribute.
Thank you. So it's a pleasure to represent the field, to share with you what AI and what synthetic biology and what AI can probably do for synthetic biology. I think a lot has been said in the first session, and actually I will say that you probably hear very similar things. It's going to be quite applicable in a lot of the earlier on speakers. I think this is really in general, I think synthetic biology, I think maybe different from drug design. It is, I mean, similar and also yet different, but really it's also, it's really a multidisciplinary field of science as well. And really, I think there's many different definitions. I'm probably putting up here just one definition here on how people view synthetic biology is really to redesign organisms, including biological parts, systems, or devices, really for useful purposes. At least that's how the field is now thinking about it. And really, you know, empowering the organisms or the cells or the systems with new abilities. And with that, of course, over the years, right, and more recently, there's really recognition that synthetic biology has really wide-ranging potential applications, cutting across many different fields, including drug development, in therapeutics, etc. And that there's really a lot more demand now, and it's really there's this recognition that it will really impact many different industries, right, from healthcare to food, consumer products, you know, basically how it can also tackle challenges, global challenges in our concerns for climate change or even natural resources. And it has been said that really, nearly at least 60% of all products in the world maybe actually one day all be biomanufactured. And so this is a very bold projection, but I would say that are we really there yet? Now, in a way, I would say that generally there's really still a lot of challenges, even though there's a lot of advancement already in the field in many years, to many aspects, right, all the way from how DNA are synthesized and how we know sequencing, all the different DNA and sequencing technologies and all the different adjacent advancements that really led to this. Technically and technologically and scientifically, a lot of researchers in the world, including ourselves over here, has been showing that it's largely possible, and there's a lot of examples out there, but it remains, I would say, largely a relatively expensive endeavor. Depending on the applications, right? If your application area is quite high value, then perhaps it's okay. It's okay to take an expensive approach. But if it's the application meant to be general, then the technology itself still remains quite a relatively expensive endeavor. And that's really because biology is actually much harder to predict than really other ways, right? In the other physical or chemical systems, and really the future, I think I would say that is really that there's a need to make it really cheaper, faster, and better, and to be on par with incumbent, because there's other choices as well. But this is also a multi-dimensional and actually multi-scale problem. And I think broadly classify that actually, whether it is at discovery phase, whether it's at prototyping phase, or is it really at scaling phase here. So one of the ways, I think, just like in drug design and drug development, people have been trying to scale through automation and standardization over the years, before actual this AI era. And so one of the ways that synthetic biology community has put together was to form Global Biofoundry Alliance. Singapore is actually one of the members, thanks to Professor Matthew Chen as well in some of these joining associations for representing Singapore, into putting Singapore in the map of this. And people have been trying to create standardized codes, trying to use automation so that you get standardized data sets, a standardized way to really create large data sets as well, robotics to really scale things. And that's really helped, right? So that's the foundation before, the recent years of, I would say, AI advancements. Of course, some of the AI advancement, AlphaFold has been, spoken about earlier on. I will not go into further details on this. And also the last few years, right, including how AI has taken into the next level, like when the release of ChatGPT and things like that. And so there's then a lot of advancement, right? I quote this AI researcher, who's also called a co-founder of DeepMind, and really that the world, it's really been predicted that the world will be defined by two core technologies in the future, right? AI and also synthetic biology. When together, it's always predicted that we will really create a new frontiers in humanities in the future. And Eric Smith also, I think yesterday, published an article in Time, also predicting that we should be ready for the ChatGPT moment in biotech as well. And that's really, I think there's a lot of interest in this area. But I think the one of the main things is that there's still need for a lot of requirements for really AI to flourish. And the need for, I think has been also shared earlier on in another earlier section, is that the need for organized curated data sets and also the ability to actually have those data sets. But if we look at it later on, I think in my others, and also the type of approach that you can build models, there's also multiple different approaches right now. I think there's also another layer of a challenge, which is really how do you then integrate different types of data sets, but even different types, even then is also not really straightforward. So if you think about how synthetic biology from end to end, what usually happens is that you usually have a problem, right? You have a need. Therefore, you select your target, and then you design, you engineer, you optimize, you scale, and you finally produce a product you want. And that, if you think about it, AI can really be impact in every stage of the way. But the types of data is actually different. The types of experimentations are also different. How do you go from one aspect of development and then getting the results to the end, right? And almost similar to drug development, when you first identify a target, all the way to reaching the clinical trials and then really getting the patients and solving the problem. So I've divided that really the advancement maybe in different areas, including the discovery phase, prototyping phase, or even the scale-up phase itself. Yeah, so then there's a lot of methods that's been developed to accelerate prototyping. I think that's really including things like enhancing it together with automation, generating large data sets, and then creating, biophysical features and machine learning models to really accelerate some of this. And other methods really include, the similarly, right? It's a local work by a professor Winston and Trelopu here, that they also create, automation, right? To really create large data sets. And because that's curated, you can then use feature correlations and machine learning to really do predictions here. And then the similar fashions then is also then applied to areas like, enzyme, protein engineering, and then creating predictive models and to do the targeted specific areas of prototyping. So that discrete manner, I think, has been shown in many different areas. And our contribution, I think, recently is really also about developing different types of model, different approaches for enzyme engineering. I will not do too much into it. I think later on, Winston will also share a bit more on this. And it's really things like, how do you integrate different types of computational modeling, different types of multimodal, so work with Sebastian and different team and how fun as well in this. And also then work into using even more generative AI approach that really needs to go hand in hand with experimental aspects as well. And so the list, I think, continues even in this. I think work also in BRI in terms of using system biology approach and integrating with AI, right? Integrating with building different types of models and things like that to really optimize for metabolic engineering. So I think that list continues, and whether that translates and in the other aspects of bioprocess development, I think you will also see very similar patterns going forward in this, right? To integrate different types of machine learning models with conventional mechanism-based methods to really predict, and interpret the results. And of course, even this very nice review here, and then people look into new types of integration to really gain new insights. I think that's where the next frontiers will really be. And even in discovery as well, right? I think in the discovery end of the things, it's like, where do you start? How do you shortlist? That's also is really more about, there's really some large data sets going around, like including things like BGC, which is empowered by a lot of sequences already a lot of things being sequenced in the world. There's also molecules database, and then methods to do predictions and mining. But I think one of the things is that when you combine different types of data sets, one of the key challenge could really be the disparity between the amount of data and the ability to generate the equal number of data, right? So one of our contribution recently was really to how can we expand a data set to be more equivalent to genomic sequences level, right? In the millions type, because sequences can be in the millions or billions right now, but really actually characterized molecules are in the hundreds of thousands. So there's really orders of magnitude of differences in a sense right now. So I think I'll just end off by saying that, yes, in a discrete manner, there's really a lot of advancements, and AI has already been alluded to and also applied. People have tried to apply AI in a lot of different aspects of the developmental phase in synthetic biology. But of course, further advancements are really needed across this developmental chain. And especially integration, right, from one end of the discovery phase all the way to the scale up and bioprocessing phase as well. And I cannot agree more with the previous speaker that really the ability to work together with the multidisciplinary thing almost on a day-to-day basis, I think is quite key to this. Thank you.
So thank you very much. I think you see a lot of similarities in biomedical sciences when you have cells, you have proteins, you have organisms. So there is a lot of learning across the fields. And I want to move quickly on to Winston to give us another brief description on some of the synthetic biology aspects. I think that goes more towards protein design. And that's what we have been discussing before. We wanted to get the structure from known human proteins in the first section. And now we're talking about creating new synthetic proteins as well. Winston.
Hi, thanks a lot, Sebastian, for having me. I'm from the Bioinformatics Institute. I've joined appointment in the Institute for Sustainability, Chemicals, Energy, Environment. So the lab, we do a lot of molecular innovations and AI integration. So today I start off with, okay, that's a lag. Two quotes. So one from Jensen Huang, the CEO of NVIDIA, the biology as an opportunity to be engineering. And, as we have seen, the synthetic biology definition has the word engineering principle from it. So in my talk, in the next few minutes, you will realize that I take a very engineering perspective to this, rather than talking about the computer science and the models. And the main part that I want to emphasize on is compounding on the benefits of the previous years, meaning actually start to learn things from the experiment. So I think we all know that there are probably two modes of engineering here, sequence and structure. So I'll skip through that and go straight into the engineering, which is like self-driving labs. We are all obsessed with self-driving cars, but self-driving labs obviously is going to be the future in the next thing. But one of the main things is that you need an agent and an environment, right? So you need to be able to learn to use machine learning to learn the sort of protein fitness landscape. And in this case, this is where I pay tribute to all the speakers who come before me. We deploy a lot of your models to actually understand the different properties of the protein and to predict the properties of protein. But engineering is not science, right? Engineering is a lot about it is about compromise. We want a protein that is heat stable, work in this pH, and yet still has reasonable amount of activity that you can produce at a reasonable yield. So how do we do that? I think previously in the RNA talk that we have, you talk about how do we prompt? How do we, navigate this landscape? Here we have we are lucky enough to have the environment do the prompting for us, right? Essentially, the environment is the one that actually prompt and allow you to actually navigate this entire landscape. So from the engineering perspective, this is very exciting, but a lot of it is all about odds. How do you actually get winning odds? How do you improve your odds of actually getting something out of it. So over here in this slide at the top left-hand corner, you can see that what are the typical setup of such an engineering system is that you need to have an agent, you have an environment, and the environment typically in terms of protein engineering, you want to assemble the design, and then you run the experiment, a biochemical characterization, and then you feed it back. And then you try to sort of learn what is the ideal path, essentially getting an agent to play the game of Go, but for protein design to know which is the next best step to take so that you can explore most of the space of the protein design most effectively and actually give you a better chance of getting a protein that you want. And at this point, I will effectively summarize the strategy that we sort of use, or in general, most people can think of is that you can use sequence as a starting point. So you want to explore sort of the sequence embedding space of all the models and the large language models that you have, or you can use structure as a starting point, right? You realize there are certain domains that you are interested in and you want to change and switch it out. How do you do that effectively? How do you summarize and actually find and survey the space? Of course, you do multimodal as well. And eventually, I think at the end, I will do one part, which is to bring in about one of the biggest trends is the cost of sequencing is coming lower. And so how can we game the system so that we have a better odds if we use genome sequence library? And as Prof Yang just mentioned, metagenomics libraries are huge. They are all out there. Can we use them to incorporate to play the odds so that we have a better hit, right? So effectively, these are the few strategies I'll be going through very quickly. So the first one is protein design using sequence to optimize for it. I take a case study here. Example is efficient evolution of human antibodies. But the real figure that I want to get to is actually this one figure in the paper, where you will see that you have a full mutational space, right? So a lot of the questions when we do engineering of the protein design for synthetic biology is, are we sampling enough of the mutational space? Are we surveying everything enough, right? So here we use basically what they use was a language model to help say that, you have, covered this space. These are the good likelihoods. Maybe you want to take a chance, go to another part of the landscape, take a look, what are the plausible mutations, what's the ones that the language model suggests, and everything else. So you make it play sort of this game and navigation down a gradient so that you'll be able to get a better chance of getting something that you want. And of course, they show that this works fantastic. In our own work, I think back to what all the previous speakers say, so it took about a long time to get into a common language. We wanted to find a sweet tasting protein, right? Because this is motivated by wanting better health. You want better health, you do not want to eat sugar, you want your drink to be grade A despite tasting sweet. So we wanted a brasserin molecule. So we already know what kind of protein that tastes sweet and we want to go after. And we are now asking, can we look around, survey, quickly navigate down and find something that is more thermostable, more soluble, and sweeter, right? And so that's where we went to navigate in the sequence space. And remarkably, if you don't navigate too far away from your wild-type molecule, you get structures that look similar, you get properties that look similar, and you can start engineering and start your selection to find things that actually work better, right? But these strategies are definitely applicable across all different kinds of modules, anything that can be expressed as a language. We understand a lot of our scientific data in languages, from chemical to single cell to gene cluster. I mean, for chemical, we are obviously trying to see, playing the game of which molecule can be synthesizable. Gene expression, what the least amount of gene you can knock down to change from one cell state to the other. Biosynthetic gene clusters, as Hwee just mentioned, can you use this as Lego bricks to produce new compounds and molecules? What's the most efficient pathway to it? And it goes down from protein language, RNA, and epigenetics. It's all amenable to a general engineering strategy, right? So this is where I pay tribute again. I think it's Prof Zhang works where, it starts to get people thinking, instead of just navigating the sequence language space, can we also navigate the structural space? A lot of the time we look at a protein, there are actually certain domains that we can switch out. there are actually certain parts of it that can, change, such as adding a DNA binding domain. Will that make the polymerase more stable and everything else? And a lot of times when we do that, we want to check that the molecule is stable, and we want to do and navigate that landscape again, such that it's stable, it can still do its polymerase action, it could still do everything else that you want, and it has reasonable yield. Right. So all these models is based on the structure base, but more recently, people are also using diffusion-based models. So we are also doing that, but this is on the generative process, right? So at the end of the day, the prompt will come in from the experiments to push all of this process towards a more functional molecule. Yeah, so the brief history, I think we're running short of time. I wanted to show this from the Baker's lab. And mainly, we talk a lot about understanding how do we know one method is better than the other? For me here, I really love this statement. It's hidden in the captions that we attribute one order of magnitude of performance better than the previous to the diffusion method, and the second order to filtering with AlphaFold 2, right? So what this tells us is actually, if you can actually incorporate many different models that everyone developed into a single framework to actually just filter it and just sieve it and just navigate a landscape well enough, you can achieve reasonable amount of success. Finally, I would say we also think a lot about, I think it's more of a challenge statement. There is no doubt that AI is going to be incorporated into engineering practices. There's a lot of biotech startup that's going to do that. So the question here for me for a long time is, I have no doubt that AI is going to be a public commodity, and all these models are going to be a public commodity in the future. So how do Singapore still ensure that we have a winning odd when we do that? And this is where I would say that the trick here is to have a big enough genomic database and to be able to do functional screening on them. I mean, this is obviously one of the domains whereby one of the models where we can use to actually annotate unknown sequences and their functions. Now there's also deep learning methods to annotate all the unknown sequences that we have. And essentially what this means is that what we want is what we did here as an example, is that we look into a strain library, a library of all the different bacterial strains. And then we ask the question, we are interested in degrading plastics, right? So can we just look into a huge genomic database, fold all the proteins, annotate all the proteins, look for the ones that look similar, rank them, and actually use them as a starting point for the protein engineering process and the feedback loop with the experimentation and everything. And here we show, obviously, we managed to discover a few molecules, a few enzymes that could actually degrade plastics just as well. So at the end, I think this is my last slide. I think for the challenge and the challenge statement is essentially, for me, all these models are going to be public commodity. Everyone's going to be doing it. The question here is, the edge that Singapore has is that we are in Southeast Asia where there's amazing biodiversity, amazing genomes floating around, right? Can we use that as a winning edge to bias our AI model so that we can win the game of the synthetic biology? Yeah. Okay. Thanks.
Thanks, Winston. So we have one final talk before we go into discussion. The discussion we will also have together with some of the lunch. You can go in and out. So don't worry, we will finish everything on time. So this last talk, it was an interesting discussion. So Professor Jianshe is new to Singapore in let's say for working here. If you've heard from Winston, he was talking about sweet proteins. And we've talked about how the drug design and the targets, how this relates to the cells, to the whole organism. So there are many, many aspects of biomedical data from many different sources that relate. And in fact, he has a very, very interesting subject area that I want you to hear about, please.
Thank you very much, Sebastian. It's great to be here to talk to guys. Actually, it's a challenge to me to give a talk. Part of the reason is I'm new. I just joined last week. I barely know anyone here. And secondly, more importantly, I really know nothing about AI. So it's a big challenge for me. It's right to talk about challenge. But I just read a book last week. I had got this quote: progress is impossible without change. And those who cannot change their mind cannot change anything. So I would like to make some progress, so I changed my mind. I accept the invitation to come over to talk about my problem and would like to seek help from AI people, whether new technology can help us, myself and my colleague at Hwee. So one problem faced nowadays in Singapore and most other countries across the globe is elderly population. The large scale population of elderly people, they have problem, not health. The most essential is that they don't have enough intake of nutrients, enough take of energy, or enough take of protein. Because elderly people, because aging weakened capability, they cannot eat, and they have much reduced desire to eat. So how can we ensure they have enough food intake, enough intake of nutrient, enough intake of protein? It's really, really important, really important for elderly people. I believe Singapore, there's the same problem. In China alone, there are 260 million of elderly people. Of them, about 30 million, about 80. It's a large proportion. They suffer same problem. And this is a problem for food industry. At the moment, the food industry is produce food, one type of food for all. And you just pick up at your own risk. So our thinking is, how can we do food design in a much better way to suit the capability of elderly? So we call this as our new initiative to put forward a grant proposal next month. We call this data-driven food design to suit the need of elderly people. And the second title is capability matching. So what we say, the capability matching here. So to our thinking, we visualize. So we have so many elderly people, each individual, male, female, young elderly, medium, and very old elderly. They all different, have different capability. Whether we can categorize these elderly people according to their physiological capability. So they are normal, or they're partly weakened capability, or heavily weakened capability. And we can categorize their capability, and then we can match up by the food industry to produce the texture, to produce a food in different level of texture, or different hardness, different moistness, to suit the different capability of elderly people. And we do already have done a lot of work on the oral physiology capability measurement of elderly people. But however, we need help how to use this data to convert into a twin model, a digital twin model, this is a new term to me, to predict their eating capability. So are you in eating capability B level, so we can, food industry can produce a food texture level, say level 5 to match your capability. And from manufacturer side also, it's quite complicated, how to cook or produce food to different texture levels through ingredient, through processing conditions, or through other combined. So this is the idea. It's a challenge for us. And more, very naive popular question to some AI guys. To us, AI is a really new technology, new technique, which it seems in far distance, but suddenly I realized this morning, it's just in front of us. And to me, how to grasp the transformative power of AI, particularly for food design, food structure design, and manufacturing. What's the impact? I guess it will be coming soon to food industry, to food science design. How can we get this opportunity to adopt the AI? That's my talk. Thank you very much. Am I within 5 minutes?
You were very perfect. So in fact, welcome to Singapore as an opportunity. I hope many people will start collaborating with you. And it was also delivered to have this talk at the end because I hope you thought a bit about food and you may be hungry. We'll just take a quick look at the pigeonhole, please. So what we're gonna do is have a mixed lunch table discussion session. So as mentioned, we have some scribes that help take down notes. We had some predefined topics based on the presenter's summary. We have, for example, maybe can all the scribes stand up briefly? So my Chen here is for the discussions around target. So please find this table and her so to contribute to the discussion of the drug design on the target side. Pietro is looking at the drug component. So this could be the structures, could be the docking, the middle section, hit to lead refinement and so on. So find Pietro if you want to contribute more in this discussion. Chen is helping us from the side for finding the right patients. So I see. So can you put the results of the pigeonhole? There, you will see a lot of people are interested in Asian-specific as well as patient-specific drug response. So AI for precision medicine, choosing the right drug for the right patient. So this will also be covered here by Chen's table. We also have Amy that takes over our general topic that we haven't defined yet. So the idea was that we look at what people are putting in, what they are voting for from some of the questions that we have put in, and we'll pick what is there. So the Asian-specific and patient-specific drug treatments, they are already there in the tables. So there is one for disease X. So this is more maybe towards preparedness for infectious disease. Of course, area I also know it's very important, and AI can contribute. We don't have a specific table for that. We do have enough people in the room to cover that. And another area here that I see is the harmonized data accessible is several of the people have mentioned the importance of the data. So I hope we can get one table together for this. But we may need to have more scribes for that. So Joseph, one of my trusted staff on the spot, are you able to scribe for one of them? Okay. So let's, and we also have synthetic biology. Shan, thank you. So synthetic biology and discussion, and related to that, please go to this table. So we want everyone online. Also, if you have more questions, keep filling up the pigeonhole or vote in there, and we will browse through it and digest it and discuss with Yang and Kedar to bring it to NRF. And so I think now if everyone has some idea to which table they want to go, please also grab some food outside and try your own sensory AI on the food while discussing some of the pertinent questions. So thank you. And after that, we'll have every table presenting the summary just for a quick wrap up. Thank you. Hello, everyone. Many thanks for the contributions. That I saw a lot of discussions. So it's really nice to see you sitting together. I'm very grateful also to our scribes that were also asked to join in very short notice. So I'm really grateful that they stood up and joined to help with this. And I think it's important for us to see if we know it's work in progress, especially this kind of table discussions, are not the final refined answer. But I think it's good that we put this crowdsourcing effort into this table, and we go through our different tables, and then later we will digest it and put it into this part of the report for NRF and discussion with Yang and Kedar. So we've moved to the first table, which was about the surrounding the right target in AI and drug discovery. And Mei-chan is kindly agreed to summarize and took note in the table.
Hello. Yeah. So we come up with like 5, 6 challenges. The first one is a hot topic on T-cell epitope vaccine design. So the role of AI would be developing AI tools that can predict either from TCR repertoire data to predict the potential antigens, or the other way around, from antigen to predict TCR. I guess the TCR profiling is relatively more specific potentially. And so the potential impact would be personalized T-cell therapy. So the second challenge is on using spatial omics that would allow us to do cross-regulatory interactions that we can find these interactions that associated with clinical phenotypes, genetic, epigenetic information, like layer all these different data modalities together with the tissue disease context in mind. And the role of AI is to find robust patterns showing this association, and also look at targets, ligands, pairs that in a particular interesting pathways or cancer-related pathways as potential drug targets. So yeah, targeting the pathways and the interactions rather than individual molecules or gene. Then potential impact is the ability to predict drug response, and also toxicity prediction would be possible that can save time and cost at the very early stage, and also ensure patient safety. Yep, and the third challenge is to do large-scale imputations of omics data. For example, with the AI data imputation capability, we can profile only 10% of the data, and then we can impute the global space using AI. So this can reduce costs and then provide essential data for AI drug discovery. And the fourth challenge is to use long-read mRNA sequencing to enhance the protein structure prediction, because long-read RNA is able to inform about the isoforms, for example, to differentiate the VEGF, alpha and beta. So the role of AI is mainly to use more advanced data technology. Then the potential impact is to find more relevant targets. Isoform can be cell type specific and or cancer patient specific. So this is towards personalized cancer therapy also. Then another one is to look at frame shift related protein targets. Okay, AI role is on predicting frame shift from mRNA data, and this can lead to enhanced protein characterization. And the last one is from plant genome to pharmacology. So the AI tools can be used to screen for potential drugs using plant genome. Yeah.
Many thanks, Mai-chan. So anyone on the table wants to add something quickly, or are there any questions from the other tables? I think that's good to move on time. I think several of these examples, they're there were a lot of opportunities there. In fact, we wanted to get the speaker for traditional Chinese medicine, which also from plant genome. And unfortunately, she had to cancel one week before. Was Linda from NTU. And but there are a lot of opportunities there. So if you want to feedback more in this area, also if you want to be more involved, have other ideas, just email us, myself or anyone that you know connected. Then we'll try to incorporate it. So we move on to the second table. We were at the target, and now Pietro very also on short notice has agreed very kindly, and he will talk about the drug space.
Hello, everyone. So at table 2, we were discussing how AI can help in the drug discovery process, specifically in developing drugs. The discussion started with talking about how AI could help, specifically with late stage failures, which are some of the most costly and more disruptive to the entire process. But the discussion quickly determined that at that point, AI isn't really that helpful anymore, because you need to intervene much faster, which is actually the challenge that we identified, which is how do we integrate AI at all stages of the process, not just at the end where perhaps we're already locked into a target that may no longer be a profitable one. How do we start AI from the very beginning, from the target to the hit identification, to lead optimization, and so on. One important thing that we discussed is perhaps going beyond the scope of just looking at the drug is how do we create a multidisciplinary team that incorporates not only chemists, medicinal chemists, biologists, but also AI experts in order to have this expertise at all levels and make sure that the proper tools are being applied at every stage. One area that we identified where AI could help the most is perhaps with drug toxicity prediction, because in that case we might be able to look at previously toxic drugs for which there may be more data, in order to make sure that we don't go down the route of these particular drugs and avoid focusing our efforts on them. And finally, one challenge is that a lot of these AI models that we have developed for drug development, a lot of models in order to look at specific molecules or specific diseases, specific proteins, often end up being very specific to that particular system and may not be transferable. So we need to identify those particular models that we can use for that can be transferred to different kinds of models and different kinds of proteins, in which way we can use these models more generally. With that, I think we are done for table 2. Thank you very much for listening.
Thank you. Any immediate urgent question? You mean you wanted to ask questions earlier? The first table? Yeah, you can ask it.
Okay. Yeah. Yes. Your presentation. So my first question is, how did you use is about the machine learning on molecular docking? So how did you leverage the molecular docking in your machine learning method?
I'm not very clear in my talk, sorry, because I don't have much time.
Okay.
So I use docking partially because historical reasons. I was trained in postdoc in the docking lab. Okay. So I know how I look at docking results. So I have that experience. So here is more like a double check. In a machine learning model, there's no docking. Machine learning, we get the top score compounds.
Yeah.
Then we use docking to do double check, something like that. Yeah. So then I have a better feeling. It's like a consensus. Yeah. It's only applied to top hit, so it wouldn't slow down the speed. We didn't really, put a docking or into machine learning or machine learning to docking yet. Okay. But it could be, yeah, it could be. Okay. But I should warn you, there are a lot of noise there, so it may not be good. Yeah.
Okay. So because we have a prohibition patent on using the molecular docking in our deep learning model, and we use self-attention to find the accurate docking results within a large amount of docking decoys.
You are trying to say use machine learning to mimic docking results, or to learn from docking
in
results?
fact, we can predict, we can do screening the drug hits within the docking results. Yeah, we use the docking complex to predict the, for example, bioactivity.
So you do docking first, then you use machine learning to screen.
Yeah, screen.
Yeah.
And we find our model can extract useful information within these docking results and also find the accurate one.
There are two ways now more in the world. And okay, there are more, but two main ways: either use machine learning, then docking to check, or docking first, then machine learning to check.
Yes, yeah.
There are some people in Sweden also do that. Yeah.
So the second question is, did you validate your lead optimization result on what experiments?
I validate my 5 target hit identification results in validation. I haven't go to the lead optimization yet due to funding. Yeah, but so far I only have 5 targets tested in cell assay or in protein assay. So each of them have some hit verified. But the next step will be lead optimization.
Yes, because I also do some work on the lead optimization. But I find that it is very difficult to validate if we don't do the wet lab experiments. So most of the time we are validate on the predictions of some consensus models. So I also trying to do that by screening out some activity cliffs in the databases. So I'm trying to optimize the molecules, just one small transformation or one small part, and I found I can use deep learning to optimize to some of the highly active molecules in database.
Sure.
They didn't think so. I think So my work is on use deep learning to do some standard tasks, but found something new during the model prediction or attention.
Yes, that's great. I mean, that can be done. Yes.
Yeah.
Great. Congratulations. Yeah.
That's all.
Thanks for the question. So we have another question online from Chandra. Chandra, can you please speak it out?
I was just suggesting that with this AI, can you hear me? Yes. Hello?
Yes, we can hear you. Chandra, go ahead. It was okay.
Yeah. I was just saying that with this prediction of toxicity of drug, maybe one could also factor in data that comes from the effect of diet and microbiome on drug interactions. With all this work going on in Singapore, may be a good opportunity to add another very important parameter, which I think is often overlooked. Or maybe they do it.
Yeah, I think there are a lot of people interested. I think Singapore has a lot of critical mass in microbiome, in this whole drug discovery community. So I think definitely it's something of interest to do. Thank you, Chandra.
Thanks.
Let's go to table 3. Janice, please.
Okay, hi. Yeah, so for us, can we move the slide? Okay, yeah. Yes, this is table 3. Yeah, so we're talking about the right patient. So I think we're mainly going to focus on the first one, which is how we can stratify patient treatment with big data. So essentially what we're trying to do is we're just trying to solve a very broad problem. And so one of the big things we actually need to understand first is actually how can we integrate the data? And then after that, looking across multiple different types of modalities, because with any clinical data set, it is possible to get firstly, not only things like your patient demographics, your disease information, which is mainly coming from your EHR data, but also things like your imaging analysis, and what else did I write? And also things like molecular assays, so things like all your lab results, all your genetic testing, and so on. So we should actually evaluate all this together to be able to see which of these data sets is actually the most predictive and how are they interacting together to form a good prediction. And so one other way of extracting the features, aside from integrating the current data sets that we have, is actually using natural language processing for unstructured EHR data this is actually still a huge challenge because not all data is neatly in tables for us. So you actually do need NLP to be able to draw some of this information out, because with knowing doctors, sometimes they use acronyms or they use different ways of describing the same thing. So we actually do need to be able to structure it well. Otherwise, the process is way too slow if you have to read everything yourself. Yeah. Okay. Yeah, and then of course, the way we will look at it, it should be from a cohort. It should be cohort based so that we don't only get the data, but then we also get it along with a clinical outcome. So we exactly know what exactly we are predicting. So we have the ground truth for our machine learning. So from this, well, the main impact is that we can gain better insight to exactly what kind of data types are predictive and cost-effective for stratifying patient outcomes. For example, if we can predict everything based off patient demographics, then we do not need molecular assays, although I think it's highly unlikely. Yeah. And so the way we will measure our success is actually using either external validation or an independent cohort with another hospital. Could be in Singapore, could be another country. Yeah. And so some other stuff we talk about as well are things like in early stage drug discovery, we need to be able to identify not only the right target, but also the right patient. So for that, we actually need to understand not just the protein that we're targeting, but also things like what are actually good biomarkers and what is the use of the biomarker? Is it for diagnosis? Is it to understand the stage? What exactly is the use case? And so some of the things we can integrate these biomarkers with are things like PBPK or PKPD models. And yeah, we can actually use AI to help and identify these biomarkers and accelerate the process of building these models as well. Yeah. And so the impact of this is to create a translational platform to evaluate drug efficacy in different disease states. And lastly, one of the main interesting questions we had is, can AI help us with when our data set is much smaller? So this is actually something that a lot of clinicians actually face, especially you actually have to slice your patient data quite a bit, because when I say somebody has cancer, you have to ask, what is the cancer subtype? How old is the patient? Do they have any other comorbidities? And sometimes you have to look at the subtype of the subtype and the site that is at and so on. So each patient actually becomes really unique. And what one patient has may not be what the general population has. And so for that, when the cohorts become really small and AI requires a huge amount of data, we actually run into problems. So for that, what we actually need is very good data integration methods in order to ask, how can we bring together multiple different cohorts and link them together in a sensible way, such that we can actually extract good, significant signals. Yeah. So this would hopefully help us to build better stratification algorithms to tailor like treatments better for patients. Yeah. And yeah, then the last one was, can we discover drug candidates using the genome? And I think other groups have also discussed similar things. So we can use things like protein ligand deep learning to screen for drug candidates. Yeah. Thanks.
Thank you. I think one of the areas, we had the AI for genomics workshop too. So pharmacogenomics also is deeply rooted in that finding the right patient. So the linkage of knowledge of which SNPs in the population could lead to this difference in the drug response is, of course, complementary to what was discussed. Any immediate questions or add-ons? Then we want to move on to table 4, synthetic biology.
Okay, so I'll be presenting for table 4, and we were discussing about the grand challenges in AI for synthetic biology. So one of the first things that were brought up is that AI can be used for bioprocess scale-up, and this is a significant barrier in terms of thinking about commercialization or manufacturing of your drugs or even other products like food ingredients, for example. So one of the application areas brought up was in achieving ultra-high density cell culture, as this would enable you to reduce your bioproduction footprint, get higher yield, and therefore reduce your time to market. And the role of AI here would be to model the cell culture parameters in order to choose the right scale-up parameters. And for that, we would need a lot more bioprocess data. For example, different analytes in the culture media, the cells themselves, pH and optical density, that sort of thing. So the second challenge that we talked about is about cross-domain integration, because for synthetic biology, there is it's a very interdisciplinary area, and generally you tend to need both biological data, chemistry, as well as the engineering side of things for the scale up. And so it's about how we can better integrate all these areas or knowledge across these areas in order to go from discovery to engineering and to scale up without kind of running into problems. Because, for example, for now, some people might focus on, say, drug discovery or target discovery. Others might focus on development. And then you get this thing where when you move from one stage to another, things don't work anymore. So here, the potential impact of AI in this area would therefore be to lower the failure rates and have more successful product or drug development. So the third thing we talked about is using AI for genome mining. I know that there's already a lot of models out there for this, a lot of data sets as well. But one particular, I guess, use case that was brought up is for blood type identification. And one of this, that would mainly be to use genomic DNA sequencing to then classify the samples or your I guess it doesn't have to be patients, but also healthy individuals. And you can use that then in sort of small medical devices, diagnostic devices, that can do maybe on-site patient stratification for further treatment. And the other thing was looking at biosynthetic gene clusters. And I guess there was some talk in the earlier talks about looking at biodiversity to really find interesting bioproducts from nature that might have some economic value. And so we think that AI can be applied to that as well. And one of the types of data that you need for this actually is the functional annotation of genome. So it's not just about sequencing data, but also knowing what the data means biologically. Yes. So the last challenge is quite it's a bit more broad, I guess. It's looking at integrating robotics and automation systems across different types of data sets. Different generation of different types of data. And so the idea here is that AI can actually link up these individual systems and create like an autonomous robotic system that can facilitate really end-to-end high throughput screening. And the idea would be that then you get faster and more accurate drug candidate discovery. Yeah, thanks.
Many thanks. Do we have any immediate question? I think also the fact, you were saying also that this can be used for the drug discovery effort. It's also the synthesis of the drugs. And in more generative AI approaches to generate molecules that nature hasn't invented yet and make them synthesizable and drug-like, still needs synthetic biology to bring it together. So in fact, integration of this whole cycle will benefit all of the area. Also in the space below, of course, automation and robotics for the whole drug discovery pipeline plays a role too. So thanks again for the contribution as well. So the last table was dynamically allocated based on the pigeonhole questions of what was listed additionally compared to the ones that we've already assigned to tables. So Jose has kindly agreed and literally assigned this morning to help summarize this, but it was also typed in by Amy from Council, who has been helping us. Thank you.
Yeah, hi. So our group actually comprises of people from Institute for Infocom Research, I2R, NUS, and Bioinformatics Institute. So we actually spent a lot of time talking about the harmonized data and infrastructure for AI model development. And we realized actually you can actually spend a whole day talking about just the data alone before you even start training. Before you even start, am I going to use an LLM or what neural network type? So even if you think about data, we thought there's three almost sequential steps: collection, sanitization, and sharing, right? So collection is, how you collect the data. And all of us here, we have wet lab collaborators or we come from the wet lab, right? And before we train a model, we get, for example, single cell data in in FASTQ, FASTA, right? Very standardized data format, but the metadata surrounding that single cell experiment can be messy, may not be very clean. Is it a liver biopsy, or is it a liver biopsy that was cultured? Two very different samples. But sometimes that metadata is not inside, and if we just pipe it into a model, it might not be ideal. So the data collection, so that's why that goes on to the second point, formation of consortia, standardized methods and protocols, not just to standardize the data format, but what data is captured around your experiment, around how you single cell experiments and all that. And along that, sanitization. So pre-specified data fields. One example we discussed was, so we're in NUS here, and imagine you do some sort of experiment or survey focusing on undergrads, right? And in the age column, if you don't actually sanitize it, if you don't take a closer look at it, you realize, how come somebody put his age as 62, right? Typo, but it's inside there. So if you don't sanitize it, it's going to mess up your model. Now, all of these, for those of us who work very closely with data, this is something you do almost on a daily, weekly basis. Sanity check. Make sure your data is clean, right? And if you're talking about data sharing, for example, I think some of us are very familiar with MOH Trust, and then BII is also, developing a Biomed DA, Data Access Repository, and also SICS, right, on the other side of NUS, Singapore Institute of Clinical Sciences. If the data is not sanitized, then there's additional labor, additional burden for all of us to clean it up, which is something we should all do already, sanity check on your own end. But in terms of pre-specified data fields, for those of us who are either in consortia or administering the Biomed DA or the platforms for sharing, the data should already be sanitized, right? So that, okay, when I get it, I will do my own check, but expected that it's clean, right? So that I can quickly get on with the model training. Then the last part about sharing also consists of two parts, right? Sharing of the raw training data, which is gigabytes, petabytes, and sharing of the model itself, so that either people can update it or use it. And the model itself, as you know, all the weights are itself gigabytes large. So it's not a trivial problem. I can't just give it to you on a thumb drive ad hoc, right? We want to think about, we put, actually, we had a third point in there, I think it wasn't updated, about privacy preserving tech, especially for data sharing, right? Because a lot of the data can be proprietary. The models, of course, I think from even some of our higher ups mandate is, yes, the data is paid for by certain people, so it is not openly shared, but we share it amongst collaborators, right? So all these points that I noted, it is not trivial. It's not trivial. We need to think about it, because sometimes after all the work is done, we realize, hey, we didn't clean up before we train a model. Then we develop more already, hey, how to share, right? So all these can actually be pain points if we don't start at the start. Yeah. I think just to touch on, I don't know whether, just quickly, I think because one person in our table was actually, he's working on explainable AI. So I don't know whether anybody actually talked about explainable AI. We felt that explainable AI, of course, is important if, let's say, you go to the bank and you're looking for a loan, but then the bank's AI tell you no, and then you want to know why, right? However, for biomedical science, at the end of the day, whether it's a prediction or classifier, it still has to be tested in a wet lab. So there is a very hard adjudication from physics and chemistry. Although, of course, it's interesting from the science viewpoint, why a certain model predicted something. Eventually, it still has to go through the lab, right, to be tested. So that's just our thoughts. Okay, very quickly, I think because this was put up by somebody in the Pigeonhole about AI modeling of disease X and surveillance. I think all of us want to put COVID and the pandemic behind us, but unfortunately, it is always on the minds now of public health and government. So one thing that I can share is the AI, personally, I'm looking at forecasting disease burden using multimodal features. So not just genetics, not just mobility measures, social media measures, even Google searches, but combining it all together. So that one, I think AI is very powerful and uniquely positioned for that. Another thing I want to share is, I think the next thing, because a lot of a lot of genomic surveillance was based on collecting individual samples, right? If you have COVID-19, they'll collect a sample, sequence it, whatnot. The future now is looking at wastewater surveillance, right? And that one, I think, how AI by wastewater surveillance is, I think, a very niche, underexplored area. So just to signal that and surface that out. That's all we really discussed because we spent a lot of time talking about the data. Thanks.
Many thanks for also covering this. I saw before there was a protein-protein interaction. So in the beginning of the pigeonhole, there were 2-3 questions about protein-protein interaction. I think it has been covered partially. The protein complex part in the CAS competition, for example. There was also a similar competition called Capri that was a competition for predicting protein interaction. But it's also not limited to protein interaction per se. Maybe Yaron, you would immediately tell us it's very difficult to, let's say, drug protein interactions, but still, it's maybe something that can be tried in future. The thing also how you were showing the diffusion. Who was showing the Winston. Winston was showing. So also the Baker Lab has been designing interface, so doing a lot of work of nanobodies binding. Then the protein language model from Brian Hwee, it was also antibody design. And so protein interaction is also something in this field where AI can contribute. It's another next step. And I also know that the ITASA version for multi-domain proteins, for example, is one of the methods that you can use for that. So yeah, I want to ask again in the round. We are trying to wrap this up now. It's good we'll finish a bit earlier, but I think the discussion has been good on all of the tables. We've collected a lot of data that we need to integrate. I know the NRF team also will use AI to help with that, and I want Zhang also to come up just for a few concluding words, which could be just, saying thank you. I say thank you first to all of you for giving us your time, for coming together, for having these good discussions on the tables. A few that are still online, 200 in total, very interested in this. So it's very, very good to see the ecosystem come together. I hope many of you have met new people that you even have interest to collaborate, and maybe in these areas that are being discussed. And I want to especially thank also our team that helped me, Maple, Keith, Amy, Shan, our scribes from BI, Janice, Pietro, and Mei Chan. So I hope I didn't forget anyone, but I also thank again Kedar and Zhang, who are leading the Bigger AI for Science initiative.
Yeah, thank you. And I try not to waste of you guys' time. And, very much appreciated, the speakers today and the discussion. I really learned a lot about, what's going on here in the community for the AI for biomedical science. And, probably, I'm working on the field of structure prediction. So this field is probably one of the first, the field show the power of AI, and then later they're really applied to different topic which is covered by RNA structure protein covered by Mao, and also protein ligand docking drug discovery by Haofang and Yuguang, and also Enrico covered the target and biomarker and this omics, omics analysis. So and later is we'll talk about the this synthetic biology. So I found that there is some things in common. First of all, the biology and also different field which we really need the collaboration. So that is really the AI for science, which the initiative is set for. And also we need computing research. And that is also AI NRF pay a lot of attention, so they spend a substantial amount of, budget, which is planned to allocate for specifically for this project. And 2 weeks, next week, I will talk, Kedar and I will meet with NSCC people, and we'll talk about the resource, the GPU and CPU need by this community, especially for AI for Science initiative. So if you guys, anyone, you have some suggestion or what kind of computing power you are needing, we are needing, then you're free to send an email to me, and then I can probably really, deliver to this NSCC people. And we are also, I'm very happy to see from this talk, I see some of this field and area which the scientists in Singapore were already in the top or among the top in the global community. So AI for Science certainly can emphasize its leadership for this, this kind of area. We are also trying very hard to, or I'm happy to say some of the very promising and young faculty, and that's also what I think AI for Science initiative can support and make them grow up. So that's all we are very happy to learn. And also, so this is for today, but just like Kedar already mentioned, this is we still have a lot to follow up, including this call for proposal and the study report. So we will continuously need your support. So thank you everybody for your attention and effort. Thank you.
Thank you again. I think also NRF and the team from NRF, thank you for helping with all the catering, organizing the room. It's a very beautiful room. It's you see Singapore as a apart from San Pedro in industry, a lot of ports, but also a lot of green. And a very important aspect I want to highlight is that NRF asked us to come here to do this, to brainstorm together. I think this should also not be neglected as achievement of giving the scientists voices so that we can help shape what it makes sense to research next. So thank you again all for coming.